So I think I should continue. So 2 minutes for the 10 minutes, right? And then further 5. Thank you. So out of 100 donors, only 31 donors in blood group A showed higher titers. And the Kruskal-Wallis test for between the age group showed no significant difference, and P was 0.98. In blood group O, there were two types of antibodies, anti-A as well as anti-B antibody. So we are first analyzing the anti-A antibody results, in which 23% donor from age group A showed titer higher of 64, 12% showed 128, and 3% showed 256 values. In age group B, 5% showed 64 titer, 3% showed 128. In age group C, only 2% donors showed a titer higher of 128. And in group D, only 1% donors showed a titer of 64. Out of 148 donors showed higher titers. Wallis test showed no significant difference between the mean age group titer, and the p-value was 0.124. In the blood group O, anti-B antibody was analyzed other than anti-A, and about 70% donor from group A, age group A, showed higher titer of 64, 7% showed 128, and 6% showed 256. 7% showed from group B age showed 64 titer, 1% showed titer of 128, and from age group C, only 1% donor showed a titer higher of 64. Like out of 100, only 33 donors showed higher titer of anti-B antibody in blood group O, and there was no significant difference when Kruskal-Wallis test was analyzed for them, as the p-value was 0.48. The titer comparison of all age groups. Now the mean of all age groups have been compared, and it showed a significant difference between their mean values, and the p was less than 0.01, and pairwise comparison was computed to determine the difference among donor of different age groups. In comparison to age group A, which is the group 18 years to 25 years of donors, found a significant difference in age group B, possessing the age of 26 to 35 years, where P is less than 0.01, and age group D, greater than 45 years, with P less than 0.05, while age group C remains comparable to age group A. This is the result of the total 300 donors which we were analyzed. About 151 possessed the low titers, that is less than 1:64, and 149 possessed greater than and equal to 164 of the higher titers. This is the comparison of all age group irrespective of their blood groups. About 29% of donors who belong to age group 18 to 25 has higher titers. In age group B, only 6% people have higher titers. In age group C, 2%, and in age group D, only 0.25% higher titers were observed in the whole study. So what we have concluded that in our population, the titers of IgM type antibody are higher in healthy blood group donors who belong to young age group. And to reduce this risk, we can perform titer wherever it is possible. And the transfusion medicine staff should be aware about the potential risk of transferring such ABO mismatch product to the patient. Since titer was only performed on random donors, so we cannot make the conclusion about apheresis donation and what is the trend and frequency in them. In case of ABO mismatch plasma, elder donor should be preferred to reduce this risk. Limitation of the study. As I have mentioned, there was no apheresis donor since they contain a large amount of plasma in their product. So further study should be done to analyze what is the trend in them. Future aspect, I only work in IgM type antibodies. I would like to suggest that further work on IgG type antibodies should be done, because they are reactive on 37 degrees centigrade inside the body, and they can create higher and even severe hemolytic reactions. Then I would like to thank Team Fatmi Foundation, who allowed me to work in their vicinity, and my teacher, Dr. Saika Tabassum, and my friends who motivated me throughout the journey. Thank you.
Thank you, Ms. Fabia. It was a very nice and interesting presentation.
Thank you.
So the presentation is open for questions. If you have any questions, please raise your hands and you can ask questions. So can I ask one or two questions?
Yes, sir.
So first of all, on what basis do you distributed your patients in these age groups? Was there any specific objective of doing this, or this was just random?
As per international criteria, the papers I have followed to pursue this study, so I have divided them in the age group that were mentioned in those papers.
So do you know about any data, prevalence data available in Pakistan or Karachi about the what are the main blood groups the population of Karachi or Pakistan has?
The main group, the most prevalent blood groups he has are mostly Rh positive, and we are very low in Rh negative blood groups. The majority of blood group in Pakistani population is B, then O, then less is A, and the very minor blood group in Pakistan, Rh positive, is AB.
Okay, did you happen to meet any donor which may have any complication because of these your findings? कोई ऐसा आपको किसी ने कंप्लेन की हो कि ट्रांसफ्यूजन के दौरान जो एन-टी-ए और एन-टी-बी के टाइटर्स आपने देखे हैं, तो जो टाइटर ज़्यादा है उसमें कोई कंप्लेन उन्होंने की है?
ओके, दिस सेमी क्वांटिटेटिव टेस्ट सर ये कहीं परफॉर्म नहीं होता क्योंकि ये बहुत टाइम टेकिंग है और हमारे पास प्लेटेड इन्वेंट्री कम होती है और ये रिस्क भी रेयर है. लेकिन एज पर इंटरनेशनल पेपर्स इस तरह की केस स्टडीज रिपोर्ट हुई हैं जिसमें पेशेंट को बाकायदा ट्रांसफ्यूजन रिएक्शन हुआ है क्योंकि उन्हें ऐसे डोनर का प्रोडक्ट लगा जिसके अंदर एंटी-ए एंटीबॉडी बहुत हायर थी, जिसकी वजह से उनके ब्लड क्रॉस मैच कंपैटिबल नहीं आ रहे थे. सारा वर्कअप होने के बाद जब कुछ भी रिजल्ट सामने नहीं आया और जब साइंटिस्ट ने फिर उस ट्रांसफ्यूजन मेडिसिन हेल्थ केयर फैसिलिटी ने जब डोनर्स बुलवा के उनका टाइटर परफॉर्म किया तो पता चला वो 512 और 256 तक हायर था. और जो पेशेंट कंप्लेन कर रही थी ट्रांसफ्यूजन के वक्त कि शी इज फीलिंग समथिंग स्टैबिंग इन हर बैक वाज एक्चुअली दैट कि टाइटर हायर था, वो एंटीबॉडीज उसको अक्यूट हेमोलिटिक रिएक्शन प्रोड्यूस कर रही थी.
So I may assume that your finding will be helpful to add an extra step in the precautionary step in the process of blood transfusion.
Exactly, it will definitely be helpful for providing a safe blood transfusion to the patient who are actually transfusion dependent.
That would be great.
Yes, sir.
Thank you very much.
Thank you, sir.
Thank you, Ms. Fabian Asim. Now I would like to invite Dr. Tehseen Quds, Department of Pharmacognosy, Dow College of Pharmacy, Dow University of Health Sciences. Please come on stage and present. Dr. Tehseen Quds. नहीं आए?
Bismillahirrahmanirrahim. Assalamualaikum everyone. I'm Dr. Tehseen Quds from Dow College of Pharmacy, Dow University of Health Sciences. And my topic of research is Chamomile, an effective alternative with curative potential for anxiety disorders. Anxiety disorders are more prevalent nowadays. According to a study conducted by World Health Organization, 264 million people are affected by anxiety disorders. Women and teenagers are more affected with the anxiety. So this is the crucial time and this is the need of the time that we have to explore the herbal medicines for the treatment of the anxiety disorders, as the medicines which are already used for the treatment of the anxiety disorders like benzodiazepines, they are producing the side effects. They have high side effects. So that's why we have to look for the alternative treatment. And alternative treatment have the chamomile. Chamomile is one of the most helpful herbal medicine which is used for the prevention as well as treatment of the anxiety disorder. Scientific source of chamomile is Matricaria chamomile, which is also known as German chamomile. This is the more potent form of the chamomile which is available all over the world. Chamomile is included in the pharmacopoeia of 26 countries, in the British pharmacopoeia as well as in United States pharmacopoeia. So this is the very well-known drug. Chamomile wonderful flowers have been used anciently as an anti-anxiety and anti-inflammatory for the treatment of various disorders including anxiety, insomnia, muscle spasms, eczema, wounds, rheumatism, PMS, diabetes, fever, IBS and ulcers. So this drug was, previously it was used as an anti-inflammatory and so very effective anti-inflammatory, but now the anxiolytic activities were also determined, and that's why this is now it become also a very good anxiolytic. What happened?
ऊपर की तरफ जो है ना arrows, right and left.
अच्छा यहाँ. Active constituents. What are the active constituents that are responsible for the effectiveness of this medicine? So active constituents which are present are actually flavonoids in the appreciable concentration, and most important flavonoids are apigenin, quercetin and nuclearin. These three flavonoids are very important because they are not only the anti-inflammatory, but it's also a very good anxiolytic. Flavonoids, that includes the alpha-bisabolol and carmazolin. And the third important fraction that is known as a carbohydrate fraction, and this is known as glucoronoxylan. Glucoronoxylan, they are all the actually constituents that are responsible for the therapeutic effectiveness of the chamomile. And most important, essential oil. You all know about the aromatherapy, and aromatherapy are based on the essential oils. And chamomile flowers actually are known for the isolation of the essential oil that is used for the aromatherapy as well, and cosmetics. Anxiolytic effect of chamomile. Anxiety disorders have been identified by the presence of restlessness, panic, increased heart rate, sweating, trembling, tiredness, rapid breathing, and lack of concentration. Actually, there are various types of anxiety disorders. Okay, so that's why we are actually considering here the only anxiety disorders generally. Okay, so chamomile extract and teas are the source of apigenin, a flavonoid found to be a benzodiazepine receptor ligand, and attached to the benzodiazepine receptors in the brain proved to be a sedative and anxiolytic. So the mechanism of action is same as the benzodiazepines. A polysaccharide, methyl glucoronoxylan, was isolated and showed significant sedative and anxiolytic effects in dose-dependent manner. That is, when we increase the dose of the chamomile, the sedative and anxiolytic activity will also increase. So what are the available dosage forms that are available all over the world and also in Pakistan? These are actually chamomile teas, chamomile extracts, and chamomile tinctures. And in cosmetology, chamomile is extensively used. Chamomile, I think you all know about the homeopathy, and homeopathy, chamomile soaps are available, lotions and creams, anti-acne creams. So that's why these are the cosmeceutical use of chamomile, as well as chamomile is also used as a treatment of anxiety in the form of aromatherapy. So in aromatherapy, the essential oil is used to calm the nerves, to treat the muscle spasm, stiffness, and other symptoms of the stress. So the purpose of the current systematic review study is to highlight the significance of chamomile as a natural alternative to treat mild to moderate anxiety disorders. In this regard, literature survey was done through online electronic databases of researchers from year 2010 to 2020, including Google Scholar, PubMed, ScienceDirect, Scopus, and Web of Science. Chamomile for clinical depression and anxiety. Chamomile is not only used for the treatment of the inflammation, for the treatment of anxiety, but it also used for the treatment of depression as well, which is the long-term effect of the anxiety. So, a study conducted in 2013 from UK's University of Nottingham Medical School, researchers explored that chamomile significantly relaxed the smooth muscle fibers and blood vessels isolated from porcine. These effects were linked to the presence of apigenin, luteolin, and bisabolol in chamomile. So apigenin and luteolin are actually the flavonoids which are present in the chamomile, while bisabolol is a terpenoid which is present in the chamomile. Another study conducted in 2019 described that chamomile tea and warm milk produce significant reduction in anxiety. So in this study, actually two groups was made, and one group was on the chamomile tea and other was on the milk. So this study also revealed the anti-anxiety effect of the milk as well. So that's why both groups will produce a significant result. Chamomile for generalized anxiety disorder. Generalized anxiety disorder is a type of the anxiety disorder which is there's no cause of fear, there's no cause of worry. So generalized anxiety disorder, this drug was also affected. A double-blind randomized placebo-controlled trial conducted in 2014 proved the anxiolytic as well as antidepressant activities of chamomile extract in dosage of 500 to 1500 milligram for 8 weeks on 180 subjects, specifically in the case of generalized anxiety disorder. The second study, in 2016 at University of Pennsylvania, explored the long-term use of chamomile in GAD. GAD is the most prevalent form of the anxiety in clinical practice. So in this study, 93 patients were treated by researchers in a period between 2010 and 2015. The researchers found that chamomile group had 40% fewer anxiety relapse throughout the treatment. Long-term use of chamomile is also safe, as it is also responsible for reducing the blood pressure as well as reduction in weight. So that's why I have told you in the starting of my lecture that why chamomile is actually is significant for anxiety, because it's having the less side effects and it's actually a positive effect on the blood pressure, which is the in anxiety your blood pressure is raised. So that's why this also reduces the blood pressure and on the long term it's also actually also reduce the weight. So GAD patients prefer alternative treatment. It was also found that most patients affected with GAD generally prefer to take alternative treatments, especially herbal medicine, to mitigate the symptoms rather to rely on conventional medicines like benzodiazepines to avoid the side effects associated with them. For reduction of generalized anxiety disorder symptoms, frequently prescribed drugs are benzodiazepines, though effective for short-term treatment, but most people generally develop tolerance to them, as a result of which experience habituation and other distressing withdrawal symptoms. Conclusion, we can conclude on the basis of this study that chamomile has been found an effective treatment to mitigate the signs and symptoms of anxiety disorders and good alternative to the conventional medicines with less side effects. These are the references. These are the actually, there are so many references were there that I have actually mentioned the significant references here. So I am very thankful to the SZABIST and LIFESCICON team for presenting my research work here, and I am very, very happy to be here to give my presentation. Thank you so much.
Thank you very much, Dr. Tahseen, for such an interesting presentation, and I think this is going to be the best-selling or highly selling herb in near future.
Thank you so much.
We are going to be patient of anxiety, the situation.
Exactly in our society, more prevalence.
Yeah.
In world, 264 million people are actually affected, but in Pakistan, there is no study conducted on the prevalence of the anxiety in our society, because of the lot of the circumstances going in our society. There are a lot of anxiety in everyone. So,
okay, I have a very basic question. What is the common name of this herb? मतलब उर्दू में हम इसको क्या कहते हैं?
chamomile is actually ये German chamomile होता है और एक Roman chamomile है. Both are the species. से ये normally जो है ये हम इसको जो है वो chamomile ही
available in
yes available, it's available. Hebion काम भी करती है, इस पर Hebion और बहुत सारी herbal industries हैं, तो उन्होंने इस पर जो है थोड़ा काम भी किया है.
Okay. So the session is open for questions from audience. So if you have any question, please raise your hands.
So I think you learned a lot by this presentation, that you can also use the alternative therapy for the treatment of anxiety, everyday anxiety. So available from in the form of teas and different types of the products are available. Thank you.
So we have questions.
Thank you, Dr. Tahseen, for such a nice presentation. My question is it important to use the complete herb, or we can use either flower part, stem part, or leaf part of that herb?
Actually, the flower parts are more effective, because the high concentration of the constituents are present in only the flower part. So actually, in case of the plants, we are choosing the main parts which are actually filled with or enriched with particular types of the constituents. So the constituents which actually I have mentioned, they are actually present in the appreciable amount in the flowers. So dried flowers are always used in case of the chamomile.
Thank you.
See?
So I would like to call now Dr. Mumtaz Hussain for the presentation on synthesis, characterization, antimicrobial activity, and molecular docking study of vinylene. Substitute 1,2,3 triazoles against bacterial DNA, and he belongs to Department of Chemistry, University of Karachi. Thank you very much.
Assalamu alaikum wa rahmatullah, and good afternoon everybody. Basically I am chemist. I have done PhD in chemistry as well, and you people are belonging with bioscience or life science. So a strong connection of life science with chemistry as well. We can say chemistry is like a bridge between physical science as well as life sciences. So here my topic: synthesis, characterization, antibacterial activity, and molecular docking study of vanillin. Synthesis, characterization, antibacterial activity, and molecular docking study of vanillin derived. Vanillin derived 1,4-disulfitidine-1,2-triazole as an inhibitor of bacterial DNA synthesis. Basically, these compounds are heterocyclic in nature. You all are familiar, and you are still in bioscience from chemistry and inorganic chemistry as well as organic chemistry. So there is introduction, experimental, and result in discussion, conclusion, and there is future perspectives. Now graphical abstract of triazole compound. This is my published work in Helium. Okay. Now triazole, basically why we are using or why we are synthesizing triazole, and basically this compound is heterocyclic compound. As you are familiar, heterocyclic compounds, as you are familiar from the heterocyclic compounds are those with carbon and other element is available in them. Just like you are familiar from pyridine, furane, thiophene, so on and so forth. There's a list of heterocyclic compounds are available in our literature as well as in our other resources. Here, triazole compounds, robust aromatic heterocyclic compounds, because these are stable one towards the climatic changes as well as environmental changes, survivable, stable to thermal decomposition, as we are familiar from our climatic changes and global warming. So on the prescription of the medicine, keep your medicine below 30 centigrade and or temperature of this environment almost more than 30 or more than 40 centigrade. So that's why these are stable to a thermal decomposition, not decompose easily on this temperature. Stable to oxidative reductive process, you are familiar from oxidation as a reduction. When they react with oxygen or moisture, they react and they lose their activity. So that's why they are very stable towards these harsh conditions. Building block of many pharmaceutical drugs. So that's why you are familiar from omeprazole, Prozac, Reulane, so on and so forth. Even though you are familiar from paracetamol, from the different drugs, from the different medicines, building block of many pharmaceutical drugs. Then purification products too easy to travel towards pH. So that's why they are stable towards acidic pH as well as basic pH. You are all familiar from the pH, and so majority of medicine may decompose their activity on acidic pH as well as basic pH. So that's why this is the range between 4 to 12. They are stable in this range. Now this is the triazole and biological significance of some compounds here. Cefadrixine antibacterial with activity, and other other drugs you are familiar from this one: cefixime, ceftriaxone, cefadroxil, and omeprazole, and there are other derivatives. Like there is 1,2-triazole, phenylhydrazone, fungicide activity. Then there is phenyl 1H, 1,2-triazole in insecticidal activity. These are the compounds in their skeleton. There is triazole compounds or triazole moiety or triazole class available. So that's why triazole is a bit full of biological applications. So that's why I have chosen here and I have studied there. And not only I have chosen there, there is full of literature with these compounds available in different fields and various fields. Now this is the triazole. We have synthesized through click reaction. You are almost unfamiliar from this one because of you people are belonging to the life science. This is some sort of complicated area according to you, but those here with a chemistry background, this is very easy and very simple. Okay. This is the chemic reaction here. In chemic reaction, there is azide one, this is the nitrogen, and there is triple bond. So that's why they react with one another, and this moiety is called this is the triazole. Basically, it's only 1, 2, 3 triazole, but there is 1, 2, 3, 1, 4 triazole. There is different isomers of this one. So these triazole compounds with different activities, like here I have mentioned with an antiviral agent, anti-diabetic agent, anti-tubercular agent, anti-rational, anti-cancer, antifungal, antimicrobial, and anti-malarial. So that's why this nucleus or this class is full of pharmaceutical as well as biological applications. Then there is synthesis of this one. So here in the first step, this is the vanillin. You people are familiar from vanilla, and vanillin. This is the synthetic one. When it's treated with this is the propyl bromide. Before this one, this is treated with a base. This potassium carbonate is a weak base. You are familiar from the carbonic acid. When carbon dioxide pressurized, treated with the water, you people are familiar from the cold drink. On cold drink there is carbonic acid, there is carbonated water. So that's why this is the base we have derived or we have synthesized from carbonic acid. When this treated this one, this is responsible to pick this H and form peroxide ion. Why this peroxide ion is found? For the sake of reactivity. In this way, this reactivity may increase, and this is the substitution reaction. This 1A will become formed. In another reaction, there is another series of phenol and bisphenols compound was treated with the dibromo-propane, and this regard, this again there is a substitution reaction with azide. Here 1A and 2A to 2K is formed in a series where then the final product, this one, there is catalyst. You people are familiar from this one. Even though we will form water when hydrogen reacted with the oxygen, only there is conditions needed like light, pressure, temperature, and catalyst. Even though 10 years and decades you may close in a container hydrogen and oxygen, they never react in a water formation takes place. So that's why majority of reaction needed catalyst. So that's why catalyst speed of the reaction is in our body. There is enzymatic reaction takes place. Here in this reaction there is a chemical catalyst. This save time, and there is little bit chances of one side reaction. This is copper sulfate and the sodium ascorbate. This is the vitamin C as well. And the presence of DM as the solvent, and the very tolerable, and the 100 centigrade time is almost 12 to 20 hours. Now there is synthesized derivatives here. Time is too short, so that's why I am going to skip here with the different yields. Now this is the characterization of these compounds. Here only two peaks I have mentioned with a characteristic peak. There is coupling between this one and this methylene at 5.25, and another peak of the double bond at 0.325. So this is the spectra through technology. You all are familiar from the technology because you are students of the biotechnology as well as the bioscience. So this is the another compounds. All these compounds characterized through technology like instruments, equipments, NMR, bus, UV, as well as IR. So this is the in vitro antibacterial screening of synthesize 1, 2, 3 triazole 3A2 K against gram positive and gram negative. Basically, main motto of study is synthesize compound, screen against and check their activity. Either these compounds are potent or not. So that's why on the behalf of this investigation or this hit and trial method, we may synthesize or we may obtain new life-saving drugs or precursors here. These compounds are treated or screened with a different strain, and there are the results. Some compounds show moderate, some compounds show good behavior. So that's why this behavior depends upon their skeleton as well as pocket size and interaction with the bacteria as well. So this is the another strain which have been screened with a gram negative. On the behalf of these results, these compounds almost show great results or good results, majority of compounds. So that's why on the behalf of these results, from these results we have obtained these compounds used as a life-saving precursors. So this is the molecular docking. You are familiar from the molecular docking as well. Molecular docking is a theoretical technique to understand binding mode of compound with the DNA through non-covalent interactions. So our body, drug or medicine may work on the principle of non-covalent interaction. So that's why this interaction very beneficial and very applicable in your body. If there is covalent interaction is more strong interaction. So that's why they may damage self or healthy cells also. So molecular docking, or even though these drugs, even though these interactions are supramolecular interactions, these interactions behind the chemistry, behind the molecules, these are the weak interactions. So this theoretical approach is considered the more suitable one for structure-based drug designing. On the behalf of this interaction, so we can design drug synthesis and drug approaches. So it's helpful in compound selection for further in vitro DNA binding studies. So this is the energy profile by which we have interacted, we have studied these compounds with the DNA. So overall, generally those compounds with a less energy, they may interact, their interaction is very good or very strong. So that's why here we check which compound is a more potential. So here 3J compound, oral 3J compound with a less energy and with good interaction as well. So that's why we have studied this compound further. Binding pockets and structure activity relationships are the docking study explored the actual binding pattern within the active site. You are familiar always enzyme work key and lock mechanism and interact with the active site. So you are familiar from two sides, one is major site and one is minor site. So always interact with the major site because of that it's easy to interact, and there is pocket sites. So minor sites is some sort of their problem, so that's why it interacts with the major site.
Dr. Mumtaz, you have 1 minute to sum up your presentation.
Yes.
Thank you.
Then the dark pores of these compounds. Another one is this is the docking of 3G with TMPK. This is the enzyme thymidine kinase TMP, and these compounds may use as a chemosensor. You are familiar from this one. Overall, I am going to summarize that these compounds may use as a drug precursors, as a chemosensors, as a biomarkers, and a different variant. in a different fields also. So here selected reduction of mercury, pH studies as well as concentration studies of these compounds. This is the comparative study of before and after. This is the conclusion. After that we have synthesized these heterocyclic compounds and obtained results and used as a precursors in a different fields. Acknowledge these are the supervisors that Muhammad Raza Shah, you are familiar from this one, and yesterday he had delivered a lecture, and this is my PhD supervisor. Thank you.
Thank you very much for a very informative presentation, and the floor is open for questions. So if you have any question, please raise your hands. And I think this, okay, Dr. Ahmad, please.
All right, so this is Ahmad Kani from Biosciences Department. All right, thank you, Dr. Mumtaz, for sharing your research work about triazoles that you have done. Well, I have a question that you have utilized synthesis method, chemical synthesis, and during my PhD, I've worked with biotransformation. So when we were doing biotransformation, we were told that this technique is a environmentally friendly technique, and synthesis, oh please don't go near the synthesis, chemical synthesis, it's a very dangerous and toxic technique to carry forward, especially in terms of solvents and the temperatures that you work on. So what is your opinion about that why should we not go for biotransformation and rather go for chemical synthesis as a way forward?
As we are familiar from this one, these compounds as we are familiar from solvent effect, synthetic reagents are also toxic, and after synthesizing these compounds may after synthesis we may check their cytotoxic effect as well. So that's why this is a little bit more tired procedure as well. जितना आप मतलब ज़्यादा जाएंगे उतना ही मतलब वाला प्रोसेस भी होगा, एक्सपेंसिव भी होगा, लेकिन यह है कि जितने भी डेरिवेटिव्स हैं वो हम मतलब अपनी मतलब जो प्लेजर है, कंफर्ट है, दूसरी बात यह है कि जितने भी मतलब रिक्वायरमेंट है विद द इंक्रीज ऑफ पॉपुलेशन, सो इट्स very necessary to synthesize these reagents, these compounds. Otherwise, अगर फर्ज किडनी पेशेंट को फर्ज आप बैठ के मतलब वेंटोजन क्रीम वगैरह लगाएंगे या फर्ज पर उसको मतलब कोई हर्बल दवा पिलाएंगे तो मतलब मेरा ख्याल है कोई 1-2 दिन में मतलब उनको रिलीफ मिलेगा, ठीक है। इवन तो आप उसको मतलब कोई वारन वगैरह लगा दें तो मेरा ख्याल है ऑन द स्पॉट वो बेहतर भी हो जाएगा मतलब, ठीक है। तो इसीलिए हम सिंथेसिस की तरफ जाते हैं। सिंथेसिस दूसरी बात यह कि बढ़ती हुई आबादी के साथ अब इन तमाम चीज़ों को हम मतलब ओवरकम भी नहीं कर सकते, ठीक है। तो इसीलिए हम सिंथेसिस की तरफ जा रहे हैं, ठीक है। और बेसिकली हम इनके मतलब जो साइटोटॉक्सिक इफेक्ट देख के ही मतलब क्या करते हैं, हम मेडिसिन की तरफ ही जाते हैं मतलब, ठीक है। तो यहाँ पे जो मैंने चीज़ मतलब करी है मतलब, ठीक है। तो सिंथेसिस करने के बाद बेसिकली साइंस जो वो टोटली डिपेंड करती है हिट एंड ट्रायल मेथड, इंस्पेक्शन मेथड के ऊपर, ठीक है। अगर फॉर एग्जाम्पल हमने बना लिया, बनाने के बाद जो हम उसको मतलब चेक करेंगे कि भाई इसका लीथल इफेक्ट क्या है, मतलब साइटोटॉक्सिसिटी इसकी क्या है। इफ दिस इज लेस टॉक्सिक, ठीक है, वी विल गो फॉर फर्दर स्टडीज, ठीक है। स्टडीज पे जाएंगे ना तो फिर हमें उसको मतलब अनदर मेथड पे मतलब इसीलिए हम डेरिवेटिव बना देते हैं, कोई और रूल ले लेते हैं, मतलब यही चीज़ है।
थैंक यू।
Dr. Mumtaz, I have one comment here.
हाँ बोले।
Whenever we used name of microorganisms for the first time in any presentation or article, we used to write the complete name instead of P.Aerogenosa. We have to mention first Pseudomonas Aerogenosa, and then after that you can use its abbreviated form. So that is my comment or suggestion.
Obviously हम जो है एब्रिवेशन ही तकरीबन यूज़ करते हैं, ठीक है. तो एक तो मतलब जैसे आप केमिस्ट्री से वाकिफ़ हैं कि ऑलमोस्ट हम सिंबल्स वगैरह लिख लेते हैं, टाइम सेविंग भी हो जाती है और हमारा मतलब जो है मिस्टेक्स के चांसेस भी कम हो जाते हैं, ठीक है. एक सुडोमोनास बार बार लिखते रहेंगे, जैसे फर्ज आप यहाँ पे मैं आपको सिग्निफिकेंट फिगर बता दूँ या एवोगाड्रोस नंबर बता दूँ, ठीक है, ये इलेक्ट्रॉन का मास बता दूँ, 31 बार आप जीरो लिखेंगे, देयर विल बिकम चांसेस ऑफ मिस्टेक्स. So here we are using abbreviation for the saving time and decreasing the chances of mistakes as well.
You are absolutely right, but this is the rule of microbiological name writing, and we can't deny it that for the first time we have to mention the full name.
No, there is a full of literature with abbreviations also. Okay. यहाँ पे हमारे पास बहुत सारा literature भरा हुआ है, मतलब क्या है abbreviations हम use करते हैं. जी.
I have a question. I am Dr. Fanshe from Salim Habib University. Did you use any antibiotic to compare your results? Like you used a standard antibiotic to check the antimicrobial activity?
Superoxide sir.
In every case, in gram positive as well as in gram negative bacteria?
दोनों के cases में check करें.
Why did you use that antibiotic?
ठीक है. सर देखिए, तो हम अपने compounds को किसी reference के साथ तो मतलब check करते हैं, ठीक है. If there is reference, so that's why we can check their activity. Either this compound is more potential than this references are not. ठीक है. अगर अपने उस reference से मतलब कोई antibiotic किसी drug के साथ या किसी medicine के सामने मतलब हम उसको check करेंगे, अगर वो मतलब क्या है बेहतर एक्टिविटी दे रहा है, ओके. अगर नहीं दे रहा तो हम उसके ऊपर मजीद ट्रायल करेंगे, ठीक है. तो रेफरेंस तो मतलब लाजमी है, ठीक है. तो रेफरेंस मेक यू मोर पोटेंट.
So I'm just curious to ask, you may be planning to check the cytotoxic effect of these compounds on any mammalian cell line?
हाँ, नो सर, मेमेलियन सेल पे तो मैंने चेक नहीं किया, लेकिन ये है कि मैंने रेफरेंस जो है जो सर को मैंने बताया कि डॉक्टर साहब को सेप्रोफिलैक्सिस के साथ मतलब चेक किया है, ठीक है. बेसिकली मैं ये आपको रेफरेंस नहीं दूँगा क्योंकि ये काम जो माइक्रोबायोलॉजी ने किया है, माइक्रोबायोलॉजिस्ट ने किया है, ठीक है. तो ये आपको मैं रेफरेंस नहीं दूंगा क्योंकि इतना मतलब बंदे को मतलब खुद पता होना चाहिए अपने काम के लिए, तो भाई ये. लेकिन सरफेस नॉलेज, ठीक है, जो आपके पास माइक्रोबायोलॉजी के पास या फर्दर जो सर बायोटेक्नोलॉजिस्ट है उसको पता होगा, ठीक है. लेकिन ये नहीं है कि मतलब बंदा जो है बिल्कुल ब्लैंक हो. हाँ, लेकिन इनकी स्टडी जो है वो मेम्ब्रेन सेल पे नहीं हुई. हाँ जी.
Thank you very much.
थैंक यू.
Thank you, Dr. Mumtaz, for the presentation. And now I would like to invite Dr. Afan Shuaib from Department of Biosciences, Salim Habib University, for his presentation.
Okay, Bismillahirrahmanirrahim. I am here to talk to you about the importance of listening to your gut. This is Dr. Afan Shuaib from Salim Habib University, from the Department of Biosciences, and the title of my topic of my research is Probiotics, the Path to a Happy Belly. And to be very specific, the title of our research is Ingested Lactic Acid Bacteria Protect Intestinal Microbial Disturbance in the Gut as well as the subsequent respiratory and gut infections of Staphylococcus aureus and Pseudomonas aeruginosa. So these are the sequence of my presentation. So first of all is a gut microbiome, the microorganisms present in your intestine, their disturbance, the overgrowth of pathogens, their infection, the concept of probiotics, what are probiotics, their mode of action, and our research findings. It's a common saying that we are what we eat. The type of food we eat, the diet we consume, it all defines, it all impacts millions or billions of microorganisms in our gut, which we call gut microbiome. So the gut microbiome consists of variety of microorganisms, including good forms of bacteria, bad forms of bacteria, opportunists, and it is believed that the human has more microbial cells than our own human mammalian cells. So if we maintain the balance between good forms of bacteria and bad forms of bacteria, we can keep our gut happy and healthy. But due to some conditions, like if there is any infection in the body, if there is a disturbance in the there is a respiratory infection, if the person is using antibiotic, or if it is using any unhealthy food, so it all causes disturbance in the gut microbiome ecology gets disturbed, and because of that the overgrowth of pathogens, and there is an infection. So there are lots of pathogens and they cause variety of infections, but don't worry, probiotics are here to rescue. What are probiotics? Probiotics are live microorganisms which, when administered in proper amount, they will give you good health, and they are present in variety of food items, like they are present in milk, they are present in yogurt, they are present in some other fermented products. So probiotics are good bacteria that can improve your health, and how they can improve your health? By protecting the body against pathogens. So these are some mechanism of action of probiotics. First of all, they they compete for the pathogens for the nutrition, they compete pathogens for the binding sites, and they do not allow pathogens to bind onto the surface if they are present. And they directly kill the pathogens by producing some proteins, by producing some enzymes, by producing organic acids, by producing hydrogen peroxide. And last but not least, they boost or stimulate or trigger your immune system, so the immune system can fight against the pathogens. So our research work is related to the probiotics gut and respiratory axis. What are the impact, what are the influence of probiotic on respiratory as well as the digestive system axis. So it says that if there is a respiratory infection in the body, or if the person is using antibiotic, there is a lots of disturbance in the gut disturbance or dysbiosis. This will lead to the susceptibility of the infection. It makes the gut susceptible to the infection, and there is a gut infection. So we use probiotic oral lactic acid bacteria treatment to treat the respiratory infections, to treat or to restore the balance of gut microbiome, and to treat the gut infection. So this is the whole scenario, this is the whole picture of our gut, our research work. Now for that we built two animal model. In first animal model we gave pathogenic aerosol infection to the animal, and in another model we gave antibiotic orally to the animal. And all the groups of animals were subdivided into two subgroups. One group was left untreated and another group received oral antibiotic treatment. After that we collected the stool sample from all the animals, extracted the DNA, bacterial DNA, performed PCR, polymerase chain reaction to target to amplify the V3 region of 16S rRNA, and then perform denaturing gradient gel electrophoresis. So this is a unique type of gel in which we create a concentration of gradient of the denaturing solution, so that the bacterial DNA from the mixed sample gets denatured, gets separated at their respective concentration. So each band represents a specific species, specific genus of the bacteria. Now these are the results. This is the lung section of the control group, and this is the lung section of the animal that infected by the S. aureus. And these are the groups which were left untreated, and this is the probiotic treatment. So the probiotic treatment has the ability to reduce the severity of the infection. This is the case of Pseudomonas aeruginosa, and the same results we obtained in case of Pseudomonas aeruginosa. As we discussed that the respiratory infection or any changes in the body, it causes disturbance in the gut. So this is a lung section, or this is a animal model that have respiratory infection. and this is a control group. So that their gut microbiome or their gut microecology gets disturbed because of the respiratory infection. And this is a case of antibiotic treatment. Antibiotic therapy has also negative impact on the gut microecology, gut microbiome, while the probiotic has the potential to restore or to revert back the gut microbiome ecology. So, gut microbiome consists of aerobic bacteria, lactic acid bacteria, coliform bacteria, and all. Antibiotic has a negative impact on all the groups of bacteria, while probiotic has the potential to restore this ecology, to this balance. In order to check what is the mechanism, we say that or we found out that the probiotic do not allow pathogens to attach onto the surface. So this is the attachment of the pathogen alone, or this is the attachment of pathogen on the surface in the presence of lactic acid bacterial cell-free extract. Okay, so this is the this is in the presence of lactic acid bacteria, or this is the attachment of pathogens alone.
On what surface you observed this attachment?
This is the cover slip method. Not we didn't use any human cell. So in order to confirm these results, we performed Calco floor staining, and the Calco floor is a dye which is used to stain EPS. EPS is an extra polymeric substance. It is like a glue, and the pathogen produce this glue around it to stick tightly onto the surface. So if there is no glue, if there is no EPS, it means there will be no attachment of the pathogen and there will be no infection. So any strategy that reduces the synthesis of, that reduces the production of EPS can also block the attachment, can also block the infection. So the Calco floor staining stain the EPS and gives blue color. This is the control, the pathogens in the absence of probiotic, and the pathogens in the presence of lactic acid bacteria. So the synthesis, although blue color is also here, but because of the light intensity, the reduction of blue color, we can observe the reduction of blue color here. अभी भी clear नहीं है, मगर यहाँ blue color है. So this is the reduction of blue color here, but the reduction of EPS is not possible in the absence of lactic acid bacteria. So the concluding remarks, we say that the interstitial microbial dysbiosis caused due to infection or antibiotic exposure can result in prolonged injurious effects on the ability of the host to resist infection. Our work reveals that the use of probiotic help to retrieve or help to restore the microbial balance in the gut. Probiotics, what is the mechanism? The mechanism is the probiotics are able to attenuate the cytotoxic effects induced by Staphylococcus aureus and Pseudomonas aeruginosa, probably by interfering extracellular polymeric substance causing pathogens devoid of colonization. These are some selected references. With this, I shall stop. If you have any questions.
Thank you, Dr. Affan, for a very informative and interesting presentation. The presentation is open for questions. Students or audience, if you have any questions, please do ask. So not asking questions means you have understood everything, so I would request Dr. Hamad कि जब स्टूडेंट्स से सवाल करें क्या समझें इस प्रेजेंटेशन के बारे में, बिकॉज these sessions are not only for just for the presenter to present the research work, but for the students to learn something, that what they have understood. So Dr. Ahmad.
All right, so Dr. Affan, thank you for nice presentation. Could you please tell us for the students that what is the mechanism of action of Calco floor white that you have?
Calco floor white basically it combines with the EPS. EPS is like a carbohydrate substance, carbohydrate moiety. It's basically a polymer. So the Calco floor binds with this polymer and gives a blue color. So the blue color indicates there is a glue here, that the carbohydrate is here, the EPS is here, or if there is no blue color, it means the EPS synthesis gets reduced and there is no infection, there is no attachment.
Do you have any questions?
The first one.
Thank you, Dr. Irfan. It's such a nice topic. I want to know what is your opinion about the use of prebiotic instead of probiotics?
Yeah, basically prebiotics are the food for the probiotics. So the probiotics are the cells, are the microorganisms, while the prebiotics are the food. So if you provide food to the probiotics, definitely they will grow. So the prebiotics are very important. So if there is no food, there is no prebiotic, there will be no probiotics. So prebiotics are much more important than probiotics.
Any questions? If there are no more questions, I will ask one question. So, do you recommend any common or general probiotics for normal use?
The problem with the probiotics is strain dependent. कि वो एक human में like वो मेरी body पे काम कर रहा होगा, but it won't work in your body. So let's suppose lactic acid bacteria जो we use in our study, lactic acid acidophilus, lactobacillus acidophilus we use in our study, it worked on my body, maybe it won't work in your body, on your body. So it depends on the species. It depends on the species of the probiotic.
And how to find out this?
Try, error, try, error.
Go to the lab and go with culture.
The answer is like आपने एक common सी example है कि जब भी किसी को stomach की complication होती है, we use as yogurt, सही है? But yogurt हमेशा हर किसी को काम नहीं करता. तो yogurt basically is a probiotic. Yogurt के अंदर live cells हैं, it's a probiotic. और yogurt के जो अपने carbohydrates, moieties हैं, these are the prebiotics. तो वो किसी individual पे काम करते हैं, some individuals पे काम नहीं करता. जी.
So we have one question.
Okay, so my question is, are there any significant side effects to these probiotics?
So far we didn't find any side effects. Like in the research articles, we didn't find any side effects of probiotics. And none of this study declared that the probiotics gets harm in the body, like it produces any harm in the body. None of the research articles says that. Sorry?
But we can say that they are not always beneficial.
They are actually beneficial, but maybe some side effect.
Benefit है यहाँ पर जो युगाड़ है.
No, okay, come.
एक बंदा जो है हमेशा मतलब उसको मतलब कि लिपिड वगैरह का इफेक्ट का प्रॉब्लम जो वो तो मतलब ऑल द टाइम
नहीं, वो ये हो सकता है कि जो होस्ट है, जो होस्ट बॉडी है, वो अगर किसी चीज़ से इंटॉलरेंट है, सेंसिटिव है, जैसे लैक्टोज की इंटॉलरेंस होती है बॉडी के अंदर, तो वो प्रोबायोटिक का इफेक्ट नहीं है, वो उस मिल्क का जो उसकी कॉन्स्टिट्यूएंट्स हैं उसका इफेक्ट है, प्रोबायोटिक का इफेक्ट नहीं है. प्रोबायोटिक्स I didn't say never produce any harm, but research articles didn't say anything about the.
Dr. Irfan, I have one question based on your previous responses, couple of responses. So don't you think it's too gimmicky to say that probiotics do work? Because I mean, for every answer you have almost said, I'm not saying that this does not work, but for some it works and for some it doesn't work. So probiotics is some kind of a gimmicky thing according to me. I mean, isn't it?
According to me, it's not like that.
And why is that?
Why is that? इसका आंसर मैं इस तरह करूँगा, like the probiotics are beneficial. Okay, the probiotics are beneficial. It's a truth thing, right? It's commonly accepted that probiotics are good. But what strain is good for your health? What strain is good for your health? It's important, right? जैसे मैंने अभी I discussed earlier कि lactic acid bacteria, lactobacillus acidophilus work मेरी body पे कर रहा है, but it won't work in your body. Maybe bifidobacterium, some other probiotic work in your body. सही है? That's the answer.
Okay.
I would like to add one thing that he already mentioned in his initial slides, that actually we have to maintain the balance of good bacteria and bad bacteria. So this is important actually. So we can't say for sure that bad bacteria will always harm the body, right? And the good bacteria has to be there. But if things are in balance, I think you will not get any symptom, or you will not get any bad feeling. So if your gut is healthy, so it means there are both bacteria present there, and they are somehow interacting with each other in a happily manner. So they are in balance, you are going to have happy, healthy and dancing belly.
I want to add one more thing, one point I want to add. Like we say that the gut microbiome के अंदर जो है bad bacteria भी हैं, bad organisms भी हैं, bad bugs भी हैं and good bugs भी हैं. But bad bugs जो है वो एक healthy individual को infection क्यों cause नहीं कर रहे हैं? Because they are opportunists. So जैसे ही body का infection, body के अंदर immune system या उसका homeostasis या उसकी normal physiology disturb होती है, then they get the opportunity. These opportunists get the opportunity to cause the infection or to cause the harm.
Thank you. We have another question. Please introduce yourself very briefly. And this will be the last question because अब समझ आना शुरू हो गया सब लोगों को topic. I think.
My name is Mohammad Wajiuddin. I'm from BS Biotechnology DAO. And my question is कि आपने कहा कि कुछ के लिए it's beneficial and कुछ के लिए नहीं. तो why do you think it's okay to invest on it? Invest your time and your money over probiotics. अगर ये इतना ही personalized drugs type है, why not go for something general जो हम सब पे use कर सकें? और एक और कि अगर if it's good for someone and bad for someone, तो क्या इसका कोई genetic reason है, like कि ये आपका strain इस area की population के ऊपर work करता है और इस पे नहीं करता?
A very good question. So अब क्या होता है? अब we research work, we just focus on the group of probiotics. We never use a single probiotic. In my study, we also use more than one probiotic. So we use a combination of probiotics. The first question first question आया था prebiotics related. So now we are using the combination of probiotics, combination of live cells, different strains of probiotics as well as prebiotics. So the combination is symbiotic basically. So the research work, the new research, they are always focused on the symbiotics, not on the probiotics alone.
I would add one thing, that probiotics are actually, that they are not only for GNT helpful for GNT, they are also helpful for UTIs as well.
Exactly.
And one more important example is Trendmax. Trendmax is equally effective for all the, for all the urinary patients. So
Yeah, the broad spectrum है probiotics, so just for this gut के ऊपर नहीं है, but our work was related to the gut.
effective or not, Trendmax का result हमेशा से अच्छा ही आता है और वो काफी लोगों को ठीक करते हैं.
Thank you.
Okay, thank you very much, Dr. Fan. It was very wonderful presentation and discussion session. And the popular one too, I think. So, Mohammad Wajiuddin from Dow College of Biotechnology, Dow University of Health Sciences, for his presentation, please.
Assalamu alaikum. This is Mohammad Wajiuddin and I'm from final year BS Biotechnology at Dow College of Biotechnology, and this is the title of our project, rationalizing the potential role of erythropoiesis, erythropoietic regulatory genes like PRB, GATA1 and FOXO3 in beta thalassemia major patients. So what is erythropoiesis? Erythropoiesis, we will start from erythropoietic stem cells. Erythropoietic stem cells are the basic and the fundamental units that initiate the process of erythropoiesis. And erythropoiesis is responsible for making RBCs. RBCs are made by this process, and I don't need to tell you about the RBC function and importance of RBCs. Erythropoiesis is an important process that is regulated by several regulators like GATA1, FOG1, PRB, GATA2 and FOXO3. So GATA1 along with the PRB, which is the PRB, along with the elongation factor E2F, is very crucial for the proliferation and differentiation of erythrocytes. And FOXO3 is responsible for regulating the oxidative stress which is produced during the process of erythropoiesis. So we have also studied about the polymorphism of FOXO3. So there are several types of polymorphism found in FOXO3, but we have studied about the single nucleotide polymorphism of GLE. So what is beta thalassemia? Beta thalassemia is a inherited disorder, congenital disorder, and it is the second most common type of hemoglobinopathy found in Pakistan. It is caused because of the imbalance of alpha and beta chains in the hemoglobin. So about the prevalence of beta thalassemia, over the globe, the 11% of beta thalassemia patients are only in Pakistan. There are two of the genes which are responsible for making beta globulin chain and alpha globulin chain on chromosome number 11 and chromosome number 16. There are a number of mutations found in beta globulin chain, like 200 almost, which are responsible for beta thalassemia major. And this beta, in this case of beta thalassemia, the excess amount of alpha chains are formed, which are responsible for binding with this HSP70. HSP70 is synthesized and then it is localized in the nucleus. But because of the presence of extra or excess alpha globulin protein, it is localized in the cytoplasmic region. And because of this dislocalization, the functionality of GATA1 is reduced. And GATA1 is very crucial for the proliferation, as I mentioned earlier. And this PRB is phosphorylate because of the oxidative stress produced in the beta thalassemia patient, and FOXO3 is also reduced. The expression of FOXO3 is also reduced in the beta thalassemia patient because of the ROS production and iron overload. So the homeostasis is quite very important in every system, in the system of hematopoiesis, for maintaining the impairment between regulators leads towards the various pathological condition. So we have found out that these regulator and markers, they can be used as a therapy in beta thalassemia in future. Our objective was to analyze the expression of GATA1, PRB and FOXO3 in beta thalassemia patients, as well as in healthy individuals. And we also find out the potential impact of FOXO3 polymorphism in beta thalassemia patients. So this was our methodology. We have collected 100 blood samples. 50 of them were healthy controls and 50 were beta thalassemia patients from Hosseini Blood Bank. This was a consent form and questionnaire. We have extracted RNA by using triazole method, and then this RNA was reverse transcribed into cDNA, and this cDNA was used for qPCR analysis later. And DNA was extracted using the salting out method, and this was used for ARMS PCR. These are the results we have found out. The normal hemoglobin level in male is approximately 13 to 16 grams per DL, and for female it is 11 to 15 grams per DL. But in beta thalassemic patient, it was 9 in male and 8 in females, 8.5 in females. This was the body mass index of the patients, and we have found out that the group with lower age have more stable body mass index BMI, but as the age was increasing, the disturbance of BMI was more and more. This was the blood group analysis, and we have found that blood group O was more common in beta thalassemia patients. This was the results of arm PCRs of the healthy individuals, and we only have find that one of the patients or one of the healthy individual had this G allele polymorphism. Rest of all of the patients and healthy were normal. This was the frequency data. Only 2% of healthy had this polymorphism of G allele. Rest of them had the normal T allele. This was the expressional analysis by using RT-qPCR, and the level of all of the FOXO3, GATA1 and PRB was reduced. The reduction of FOXO3 was because of the oxidative stress and iron overload in the beta thalassemia patient. PRB was because of the phosphorylation, which was because of oxidative stress, and GATA1 was reduced because because of the excess alpha chains were responsible for this. Excess alpha chains reduce the excess alpha chains bind with the HSP70, and this HSP70 is reduce the function of GATA1. That's why GATA1 was underexpressed. Ineffective erythropoiesis has been connected to the beta thalassemia pathology, and it is responsible for several secondary problems. GATA1, FOXO3 and PRB were the more important erythropoietic regulators, and we find out that they were highly underexpressed in beta thalassemia condition. Thank you. Any questions?
Any questions, please? If you have any questions, please ask. Any questions? So, if we don't have any questions, means that your presentation was very well taken. सब कुछ समझ में आ गया सब लोगों को. Thank you so much. So our next presenter is from Department of Biosciences, Salim Habib University, Ms. Sana Mayani.
ये तो सारा जो इस पर तो खैर बहुत खेल सकता था जीन एक्सप्रेशन पे इस पर.
सो जो ऑडियंस और प्रेजेंटर्स न्यू यानी कि थोड़ा देर से आए उनके लिए मैं दोबारा बता दूं कि हमारे पास एक आपको बीप की आवाज़ आ रही होगी. सो एवरी आफ्टर 8 minutes ऑफ द प्रेजेंटेशन ये बीप हो जाती है. इसका मतलब होता है कि प्रेजेंटर हैज़ 2 more minutes एंड उसके बाद हम 5 minutes के लिए क्वेश्चन आंसर सेशन रखते हैं. तो ये परेशान ना हो कि मेरा कोई मैसेज आ रहा है एवरी टाइम टू की जो आ जाती है. दैट इज़ हमने अलार्म सेट किया है फॉर 8 minutes, राइट. थैंक यू.
Okay, so since COVID just passed, I'm sure pretty much every one of us sitting over here found wearing the face mask absolutely horrible. I mean, they were suffocating, and since we were really not accustomed to these before, we found these really hard to breathe through them. And so imagine yourself wearing one, but now all day and every day. And on top of it, add excessive sweating, painful headaches, and constantly feeling nauseated and drowsy all the time. So in case you're wondering, I'm not talking about face mask anymore. Instead, I'm talking about something that we're all very well familiar with. And this something is carbon dioxide. And this is exactly what our future will be if we're unable to control the rapidly increasing toxic levels of carbon dioxide in our environment. So hello everyone, this is Sana Mayani from the Department of Biosciences from Salim Habib University, and the purpose of my research was to bring down these toxic level of carbon dioxide by utilizing a certain specific natural ability of plants called Phyto Sequestration. So coming on to what Phyto Sequestration is actually, well basically it is a process by which the carbon dioxide which is present in our atmosphere is taken up by the plants and the trees, and while some of it is converted into oxygen by the process of photosynthesis, most of it is actually stored in the form of carbon inside the plant's own roots and the branches, and so in doing so the plant is actually able to capture and store this carbon dioxide, and as a result the carbon dioxide levels in the environment gets reduced. So the significance of our research was that in order to decrease these rising levels of carbon dioxide, rather than opting for some expensive physical or chemical means, we were simply utilizing the phyto sequestration ability of the plants, which is completely natural, and so it makes up for a much cheaper and a reasonable alternative. So keeping this in mind, me and my team, we carried out our research with the intention of checking for two things. Firstly, to check the amount of carbon dioxide that can be sequestered or removed by the different plant species, and secondly, to check if environmental pollution has any effect on the phytosequestration ability of the plants. So our first attempt was to find out the carbon dioxide levels which are normally present in our atmosphere, and for this we recorded the carbon dioxide level at 6 different spots within the Korangi Creek, which was the area of our study. And as we had expected, the levels were much high. I mean, the normal carbon dioxide value is about in the range of 250 to 350 parts per million, but we found that the carbon dioxide levels in our area of study were about 1200, so which was really high. Our next step included the collection of the plants, and since we were looking out the specific plant species that were native to the area of our study, we were able to lay our hands on 6 different plant species which are shown here on the right, and these were champa, sharifa, sadabahar, Malaysian palm, China rose and dragon tail plant. So after that our selection of plants had been done, we took the leaves of these plants while the roots and the branches were still intact into our lab, and in the lab we divided these plants into 2 sets. So one of the set contained the clean leaf plants from which the pollution has been removed completely, since these were washed thoroughly with the distilled water, while the other weren't washed at all, so they still had some of the pollution and dust and dirt on it. So this is how we were actually able to measure the phytosequestration ability of our selected plant species. This here is a photosynthetic chamber, which is really just any airtight transparent container, and on the lid are attached the 2 sensors, one for measuring the carbon dioxide and other for measuring the oxygen. So what we did was we took 2 different carbon dioxide readings, the initial readings and the final readings. The initial readings were taken when nothing was placed inside the chamber, while the final reading was taken 1 minute after we kept the plants inside the container. So in order to determine the actual amount of carbon dioxide that was sequestered, we simply deducted our final readings from our initial readings, and the same was repeated for all 6. So this is the data that we got from our experiment, and here we were able to observe that our different plant species remove different levels of carbon dioxide from the environment. So the left column here shows our selected plant species along with their respective masses, initial and final carbon dioxide, and of course the amount of carbon dioxide that was sequestered. So we observed here that the highest level of carbon dioxide that was removed was by the sharifa plant with the value of 1025 parts per million in a minute, while the lowest was by the China rose plant with the value of 42 parts per million in a minute. This is the graph for the 2 plants, China rose and the dragon tail, and these basically show the decreasing levels of carbon dioxide over time, and by this we were able to record the graphical representation of the decrement in the carbon dioxide levels, and all of our plant species showed a similar pattern where the carbon dioxide level was initially very high, but with each second that passed these levels got significantly reduced. The table shown over here is for our second objective, which was to check for whether environmental pollution interferes with the phytosequestration ability of the plants, and the leaf type basically discusses about whether the leaf were washed or not, and from this we determined that actually more amount of carbon dioxide was removed by the leaves which were washed and from which the pollution was removed, since 388 part per million carbon dioxide was removed by the unpolluted leaves compared to the ones that weren't washed at all and were polluted with the value of only 145 parts per million. So from all this data, we were able to conclude three things. Number one, that every plant that we had selected had the ability to sequester carbon dioxide. Secondly, the level of carbon dioxide that was removed was different by each plant. And lastly, environmental pollution does actually interfere with the process of phyto sequestration, since even higher amount of carbon dioxide was removed by the leaves that were washed. So in conclusion, through this study, we were able to highlight some of the potential plant candidates which we can utilize for mitigating the higher levels of carbon dioxide, and in doing so, we can also enhance their phytosequestration ability by keeping them free from pollution. So all in all, the plants that are mentioned in our study, besides serving their ornamental purposes, can actually be used to manage the toxic levels of carbon dioxide in our environment, and in doing so, these may also help us provide protection from the various detrimental health effects we might have to face otherwise in case these toxic levels of carbon dioxide are left unchecked. So this was our whole research team along with our supervisor Dr. Affan as well, and thank you.
Thank you very much, Miani, for sharing your views about...
Sir, it's Minai.
Minai, sorry. So, thank you very much.
Thank you. Minai.
Minai, for sharing your views about ignored yet very important problem of Karachi city. मेरा बड़ा एक सिंपल सा सवाल है कि what was the cost of that leaf chamber you designed?
सर मतलब सिर्फ एक कंटेनर की जो कॉस्ट,
वो सेंसर्स
सेंसर ऑलरेडी मतलब लैब में अवेलेबल थे तो
बड़ा हैंडी सा लग रहा था वो बड़ा. Okay, so I would request the audience, please ask question even if you do not have. So we have. You can start from here.
Okay, that was really interesting. I guess I have a very general question. I'm wondering about all the trees and all the plants we have in Karachi that have already been planted. Because the pollution is present in the air, what would you suggest we do for those plants so they can sequester more carbon dioxide and we can maximize that?
In order to make the plant sequester more carbon dioxide, we have to opt for the genetic engineering applications, and that would be that we increase their stomatal density so that they may absorb more carbon dioxide. And if we come to the simpler options, then we can keep them pollution free by sprinkling them with water regularly and such.
Thank you so much.
Assalamu alaikum.
Walaikum assalam.
Very nice presentation, Sana Minai.
Thank you.
And I would just ask one question, that why you didn't take the neem, Melia, as a director? Because this is the most common tree found in all over the Karachi.
We actually selected the ones which were found in the garden of our university. So it was really easy for us to check for them.
Okay, thank you.
So we have another question on the back.
We are using more and more plants.
Okay.
You said that Sharifa plant consume more carbon
I'm
dioxide.
sorry, I cannot hear you properly.
Hello, my name is Salar.
Can you please speak louder?
Hello, my name is Salar. I'm a student of Biotics, 7th semester. My question is, you said that Sharifa plant consume more carbon dioxide.
Yes.
So during photosynthesis, it can consume more carbon dioxide. So it means during respiration, it may release more carbon dioxide. Maybe possible. Maybe I am wrong.
If it is absorbing more carbon dioxide, then?
During photosynthesis, it is absorbing more carbon dioxide, Sharifa plant. So during respiration, maybe it releases more carbon dioxide. Maybe I am wrong.
Because the process reverse हो जाता है ना?
हाँ.
Night में जब
हाँ.
is it? आपको पता है ये बात? तुमने स्टडी की है इस पे क्या नहीं?
नहीं, I don't know about that.
The answer is not yet no.
Assalamu alaikum. My name is Saida and
We have answered to that question. Let's see what discussion goes on.
Hello, my name is Mohammad Mubashir Khan. I would like to add कि कार्बन डाइऑक्साइड जितनी प्लांट के इन्वायरमेंट में मौजूद है वो वही कार्बन डाइऑक्साइड है जो वो कंज्यूम भी कर चुका है और यूज़ भी कर चुका है. तो लाइक जितनी कार्बन डाइऑक्साइड मौजूद है इट्स नॉट लाइक कि वो ज़्यादा रिलीज़ कर रहा है. अगर कार्बन डाइऑक्साइड की कॉन्सेंट्रेशन कम हुई है तो इसका मतलब यही है कि हाँ वो कंज्यूम ज़्यादा कर रहा है एज़ कम्पेयर टू वो रिलीज़ कर रहा है.
अस्सलाम वालेकुम.
बेसिकली क्या होगा कि वो जो कार्बन है जो कार्बन वो कंज्यूम कर रहा है वो टोटली बॉडी के अंदर स्टोर नहीं है, वो सॉइल के अंदर भी जा रही है. तो वो सॉइल के अंदर भी अगर उसका पार्ट चला गया है तो लिटिल अमाउंट ऑफ कार्बन डाइऑक्साइड वो अगर ड्यूरिंग रेस्पिरेशन सिक्रीट आउट करेगा. तो ऐसा नहीं है कि जितनी कंज्यूम कर ली उतना ही सब कुछ रिलीज कर देगा, वो अपने मेटाबॉलिज्म के लिए भी और अपने सॉइल को ट्रांसफर भी कर देगा.
Assalamu alaikum. My name is Saida and I am also a biotech student. मेरा सिंपल सा क्वेश्चन ये है कि आपका जो एक्सपेरिमेंट है ये आपने कितने टाइम्स रिपीट किया?
About 4 months.
4 months. और कितने टाइम्स आपने रिपीट किया है? 2, 3 कितने टाइम्स?
Repeated thrice. We
Thrice.
repeated thrice.
तो
And
एवरी
then
टाइम आपको सेम ही रिजल्ट मिला इसके जो आपने आई गेस शरीफा प्लांट के बताए थे 1025 कुछ सेम ही रीडिंग्स आपको मिली
एक्चुअली
थी?
हमने ये 3 दफ़ा रिपीट किया था, फिर ये मीन वैल्यूज़ थी ये सारी.
अच्छा मीन
हाँ,
वैल्यूज
mean values थे.
तो कौन से मेजर फैक्टर्स थे जिनकी वजह से आपके एक्सपेरिमेंट के जो रिजल्ट्स थे उनमें जो है ना वो आ रहा था? क्या वो इन्वायरमेंटल फैक्टर्स थे जो अफेक्ट कर सकते थे?
मतलब ड्यूरेशन बहुत ज़्यादा लॉन्ग था, मतलब 3 months थे, तो ऑब्वियसली एनवायरनमेंट में कार्बन डाइऑक्साइड के जो लेवल्स हैं वो भी बहुत ज़्यादा वेरी कर रहे थे, तो इसकी वजह से.
आई गेस आप तो किसी चैम्बर में कर रहे थे, वो कंट्रोल होता है.
नहीं ना, मतलब चेम्बर जो है वो भी एक तरीके से इंडोर ही है ना, इंडोर ही रखा हुआ है. हाँ, तो और फिर ये होता है कि बार बार लिड ओपन क्लोज हो रही थी, तो उससे भी बहुत ज़्यादा फ्लक्चुएशंस आ रही थी कार्बन डाइऑक्साइड के लेवल्स में.
अच्छा, इट डज अफेक्ट एक्चुअली योर रिजल्ट्स. थैंक यू.
थैंक यू.
So we can have one more question. Dr. Ahmad, you have a question?
टाइम बहुत आगे चला.
Hello, assalamu alaikum. अच्छा, my question is कि how can we use this procedure normally in our daily life? मतलब क्या मैं अपने गार्डन में शरीफा प्लांट्स लगा दूँ?
हाँ, यही पॉइंट है स्टडी का कि
यही है
हम,
ना?
हाँ यही है.
अच्छा, ठीक है.
और शरीफा बहुत आसानी से लग भी जाता है, उसके अंदर बीज भी बहुत सारे होते हैं. You just have to खाओ और फेंक दो. Dr. Ahmad, please.
जी जी, thank you Ms. Sana for your informative presentation. I have a question about the selection criteria of the plants. So is there any consideration about the classification, based on classification, like whether you choose it okay, these plants are C3 plants or C4 plants, because for carbon sequestration, this plant being C3, C4 or CAM plant, it is important to know about in the first place.
Like I said before, we selected these plants because these were very easily available to us. These were in our backyard basically because these were in our garden, and the research is still going on and we will experiment with the more plant species.
Actually, there is one technicality related to that. So if you are saying Sharifa is much more effective and there is another plant with rose which is not effective, so it is partially due to that C4 plants are always effective, and C3 plants are less so in the environment, in the normal environment. So when we are saying these, we need to have this background check or background knowledge about the plants we are using. So if you have not included that criteria in your study, that might be one of the idea to include.
Yes, I think you're right.
Just one last question.
I just have a comment, not a question, that as you mentioned, you perform the experiment in triplicate, thrice.
Yes.
So it's better to mention the standard deviation along with your mean values.
Okay.
Only then we can say that that result is significant or not. Okay.
Thank you for the suggestion.
So thank you very much, Sana, for this wonderful presentation. And we have now Dr. Tehseen Fatma from Dow College of Biotechnology, Dow University of Health Sciences for his presentation, please.
Mic, ठीक है? So, let me ask Dr. Kashif. Dr. Kashif, can you hear me? If you can, then everyone else can. Okay. So yes, I'm Dr. Tehseen Fatma. I'm from Dow College of Biotechnology. The data that I'm going to present about, it's about it's a small part of a very large study that we're conducting on the molecular basis of beta thalassemia. A part that you have just listened to Mohammad Waji. So the study, or basically the whole story, is all about understanding the secondary complications that are happening at the molecular level in the patients with beta thalassemia. When we talk about these patients, so beta thalassemia patients particularly, or any specific type of pathological conditions, we mainly target the prime cause of them. For them, in the case of beta thalassemia, we know this is the disease condition. It is a congenital condition that runs in the families, happens mainly due to the consanguineous marriages, and it is basically caused by the defective beta globin gene. The patients are usually referred nowadays for hydroxyurea treatment and certain lifelong modalities such as frequent transfusions, iron chelators, vitamin supplements, and certain dietary restrictions in as well. And that is usually the end of them. This is how the management all around the world, or like most of the management, is actually going on. For us, the study was more about what happens beyond them. Is there anything that we can do? We might not be able to increase the life expectancy of them, because practically speaking, moving towards the gene therapy and correcting the beta globin gene, that's going to take long. Practically speaking, by the time they are alive, can we increase their life quality? And for that, we need to think beyond what is happening apart from thalassemia and hemoglobin. Right? So the idea was to understand what is happening, or what are the damages that are happening at the cellular and the molecular level. So we have segmented it into different parts. This part of my study mainly talks about the oxidative stress and the management of that oxidation happening in these patients particularly. We have worked with a cohort of the sample. Again, the study is still in continuation. The data that I'm going to present in today is from the people of about 100 cohort samples. Sorry, let me be a little familiar. Oops, sorry. Okay, so when we talk about oxidative stress, I'm not going to school about oxidative stress kya hai and aapke paas kya hai. It's a basic physiological process that is happening in the body. The generation of reactive molecules, this is something that is happening as a part of the basic metabolism. Aapki body mein phosphorylation bhi hoti hai, respiration bhi hoti hai, a range of processes are going on, and a result of most of them, certain reactive molecules are being produced. These are sometimes good for us. They maintain our cell cycle and are very important in producing or providing a significant level of immunity for us. It does, or it gets bad when it gets beyond the level of management, and that is why what we call it as an oxidative stress. The stress that cannot be managed anymore. That means it has moved beyond the antioxidant capacity of the system. Since this is a physiological process, our body is very well equipped, so it provides the level of protection. Now, it's not happening in a sequential order, but because this is something happening all the time, multiple levels of protection are activated simultaneously. The first line of defense when we talk about is we are producing the antioxidants that are neutralizing those reactive molecules. So if those reactive molecules are neutralized, and by neutralizing I mean for example, the nascent oxygen that is going to be produced is converted into a more or lesser toxic form that gets to be hydrogen peroxide that can then be converted into the water and is going to be more easily accommodated into our metabolic system without causing much of the distress. So that part is basically taking care of the superoxide dismutase, which is a very important antioxidant present into the system. Once we go beyond that, the reactive molecules are also the part of not only the reactive oxygen species but also reactive nitrogen species. This is also a part of stress that is being generated, and that is mainly regulated by the NOS2. NOS2 is an enzyme that regulates the levels of nitric oxides in the body. Those nitric oxides, once present into the system and reacting with excess hydrogen peroxide, are going to provide hypoxynitrites that are deleterious equally. Another enzyme that happens to provide the protection against oxidative stress is OG1, which is a glycosylase that is a part of the DNA repair mechanism, and it provides the later stage of the defense when the damage has already been occurred, but it can be repaired before it can be passed on to the progeny cells. So I'm going to talk about these three major repair systems, or basically antioxidant or the protective against the ROS elements. This is what we call as in combination. There are other antioxidant systems and other enzymatic systems in as well. But collectively, the ROS production is going to be divided into the enzymatic carried forward by the SOD2, NOS2 and OG1, and the ROS production is also going to be the non-enzymatic one that is majorly provided by some of the major enzymes into the system, most prominently vitamin D, vitamin C and vitamin E. These are the ones that have got a very important role in providing the defense against oxidative stress. In our study, I'll be talking more about vitamin D. So when we talk about when we talk about the damages happening in the thalassemic patients are the ones which have got a very defective metabolism in terms of the iron competencies and metabolism of the iron happening. And secondly, since they are very heavily dependent on lifelong transfusions, besides being provided with the iron chelators, they end up having a lot of excess iron present into the system. Now, technically, iron in our body is the element that gets recycled. We have got a very efficient way of recycling, but that is our way of producing the RBC through erythropoietic system and then degrading that as a part of the bilirubin and excreting out of the system. As a part of the transfusion, we are actually, and the patients basically end up accumulating those excess iron, and those excess iron get into the part which we call as the Fenton reaction. That Fenton reaction basically promotes or basically exaggerates the damages caused by the oxidative stress and basically adds on to the different types of the damages that could happen. The damages that we are talking about are happening at the macromolecular levels. The ROS elements have got the potential to damage the proteins in the body, the lipids of the system and also the nucleic acids. And the most deleterious one gets to be of the nucleic acids because they have the tendency to be passed on to the progeny cells and are very important cause of the transmissible cancers into the progeny cells as well. Sorry. Is it me doing something wrong? Okay. Okay, so in this case, since we are going to talk about the vitamin D as a part of the regulatory element in the ROS, the levels of the vitamin D are managed by a range of different resources depending on how much we acquire them, but the levels of vitamin D are also very much connected with the vitamin D receptor protein. It's a protein which gets activated and then further activates a range of other transcribable elements upon its binding to vitamin D as a part of its response element. The studies have found that vitamin D is a highly polymorphic genomic region, and there are plenty of them which have got very significant role. We have targeted the FOC1 polymorphism in here, which is the part of the exonic system of the VDR gene. This is of most of the polymorphic regions of the VDR. FOC1 gets to be one of the very significant ones because
it basically affects the length of the protein, sorry, is that me? It basically affects the length of the protein and affects the functionality of the protein as well. For example, if you have got a T present in place of a C, which is the defector gene, you're going to have a slightly protein which has got 3 amino acids bigger, and it has got a lesser transcriptional activity as compared to the native form, which contains a CG present in there. So having this aspect of polymorphism is going to affect the functionality of the VDR polymorphisms and is going to affect the functionality of all the downstream proteins that are going to be in turn regulated by the VDR gene. We are also targeting the polymorphisms of the NOS2. This is something that we have checked in as well. Although the polymorphism of the NOS2 is present in its intronic region, it's a part of the open reading frame, and the polymorphisms of the NOS2, despite not being the part of the coding region, the studies have shown that it affects the functionality and affects the activation of the protein directly. One of the proteins that we are going to target in our study is HUR, which is human antigen R. This is the protein which is an RNA binding protein and affects and regulates the functions of most of the antioxidants, including SOD2 and NOS2, that is the part of our study. SOD2, as I mentioned, it is superoxide dismutase, and it's mainly responsible for the conversion of the nascent oxygen or the reactive oxygen molecules to the hydrogen peroxide. Not spending much about the statistical analysis, we have already known that it is one of the very densely populated, which have got a very high positivity rate for beta thalassemia, and one of the very important reasons which we have further checked in our study are basically the consanguineous marriages. So we are still very densely populated with beta thalassemia individuals. The objective gets into be, we are going to analyze the oxidative stress in these patients, and we're going to check in the effect, we are going to check in the effect or frequency of the polymorphisms in our population of our respective genes, and we're going to check in if these polymorphisms affect the functionality of that specific proteins. We're also going to check in if the vitamin D levels and ROS levels, what are the comparative levels of the beta thalassemia patients in our population as compared to the healthy ones. That was our basic methodology collection center. Our collaborators for this study have been the Hosseini Blood Bank. So we have collected all our patients from them. The healthy ones, दोस्तों को पकड़ के खून से खून निकाल लेते हैं. It does the job. Beta thalassemia volunteers, again, they were volunteers. We did not provide them any sort of benefit. Samples collection, transportation, extractions. For the polymorphism analysis, we followed the technique of allele-specific PCR. You can also know them by the method of ARMS PCR. And for expression analysis, the RNA was extracted and expression profiling was done by the delta-delta CT method by comparing the transcript levels and comparing them with the housekeeping gene and then with the healthy controls. Talking about the results, the allele-specific PCR, as I said, the results of allele-specific PCR, it's one of the very commonly practiced, it's very cheap and equally reliable, well-reported technique. It is much, much cheaper than going toward the Sanger sequencing, a little tricky to practice though, but you get in the results in the form of a gel electrophoresis. You need to optimize the PCRs using either 2 sets of reverse or either 2 sets of the forward primers and resolving them on a higher percentage gel. So the idea gets into be, because we had 2 reverse primers and they were different by the presence of either of the 2 nucleotides, if you get the band on the C or T both, the population is considered, or that person is going to be considered as the heterozygous. Having a band only at T would mean the person is homozygous for the T allele, and a band present at C only, that is in the 3rd block, would mean the person is who's going to be homozygous for C only. That was done for all the 100 subjects that were studied during the population, and the results were calculated in terms of that. So we end up calculating how many heterozygous do we have and how many homozygous mutants and how many homozygous wild types do we end up with. In terms of VDR, what we have found in the patients have got a more frequent T allele present in them. And as I've said earlier, presence of a T allele at that specific location that we're studying, the presence of T allele is going to end up with a larger protein, making the protein less functional and contributing towards a less efficient vitamin D receptor protein. Vitamin D receptor itself is a transcribable element of a range of different downstream proteins, so that affects the metabolism marginally at a very longer run. So yes, our patients, they have got T alleles and they have got lower levels of vitamin D levels. We have checked that through ELISN as well. I'll come back to that in a minute. Talking about the NOS2, and again, the data is read in a similar way. Having the band on either C or T gets to be the homozygous ones. Having the bands on both the wells going to give you a data of the heterozygous ones. And the study was represented in the form of genotype and allele frequency. What our study was talking about is the patients of the beta thalassemia patients usually have got either they were homozygous for C allele or T allele, whereas the healthy population was the one that was more frequent for the presence of the heterozygous ones. So heterozygosity is providing some sort of natural protection in providing or maintaining the ROS levels. That we would talk about in a minute as well. In terms of the expression profiling, what we have found in is the patients have got a higher NOS2 levels as compared to the healthy ones. Having higher NOS2 levels would mean they are going to produce more nitric oxide contributing towards the nitric stress, or collectively to be called as oxidative stress. The patients of beta thalassemia had much lower levels of OG1. Now what was OG1 doing? OG1 is one of the proteins that was supposed to correct the DNA damage. So we have introduced more damage and we have not corrected. We have left it uncorrected. So all the damages that we have inferred, the DNA damage or the part of that is going to be transferred to the progeny ones, and all the DNA damage being accumulated throughout the life of the cell has got a higher tendency to end up as the part of a cancerous cell.
So we have entered our discussion time.
I'll try to be as quick as I can. The same happens with the SOD2 and HUR. SOD2 and HUR are downregulated in as well, introducing or contributing towards the oxidative stress. VDR I have already talked about, because the patients have got the measure of the T allele, the levels or the expression levels are going down. The vitamin D levels are the ones that get to be contradictory as compared to what we expected or what is actually should be reported as a part of the literature. The patients in general are expected to be very low in vitamin D levels as compared to the healthy ones. The metabolism is already very much affected. However, this does not get seen to be the case, because as a part of their maintenance regime, they are supplemented with higher levels of vitamin D. So whenever, although we have checked on their serum vitamin D levels, they might not be the true representative of the vitamin D being produced in their bodies. The data from the Fox assay again tells in, or basically confirms, that the patients that we are talking about have a very good serum high oxidative stress levels in the body. So if I like to conclude that our patients, we have checked in, they have got a very high the reactive oxidation stress present into the body. That is going to be contributed furthermore. They are going to produce a lot of reactive molecules as a part of their natural metabolism, their physiological metabolism. It is being stressed furthermore by the increased level of NOS2, so that adds on to the NOS levels. It's left uncorrected by the lower levels of OG1, and that increased level along with lower levels of VDR polymorphism is introducing a DNA damage or multiple DNA damages in every round of the cell cycle, and puts our patients at a higher risk of secondary complications, including multiple myelomas. So the basic essence or basic rationale of the study was to actually consider the patients as for secondary pathological conditions apart from just treating them for iron chelation therapies. Thank you.
Thank you, Dr. Tassim. It was a wonderful presentation. As we are running out of time, we'll have only 2 questions. If we have any questions from audience. You have questions rather than comments. Agar questions hai to please do ask, right?
Yes, please.
Assalamu alaikum. This is Mazahir Hussain, student of MS Biotech. मैडम मेरी क्वेश्चन ये है, आपने कुछ जीन्स की जो है ना बात की है, बेटा सेल्स में उसे मल्टी म्यूटेशंस कर देती है और बहुत सारे म्यूटेशंस ऐसे हैं जो जीन्स है जो पाकिस्तान में अभी तक आइडेंटिफाई नहीं हुआ है। तो मेरा ये क्वेश्चन है, जो आपने जीन्स जिनके बारे में बात की, ये पहले पाकिस्तान में आइडेंटिफाई या फर्स्ट टाइम आइडेंटिफाई हुए?
First thing is, it's not about the identifying the gene in a population. It's all about identifying a specific polymorphism in our population. These polymorphisms have not been studied in our population. In fact, the prevalence of SOD2 in beta thalassemia is still not very much considered in it as well. So these polymorphisms have not been identified.
थैंक यू मैम। अच्छा, जो आपने जीन के बारे में बात की है, क्या सिर्फ स्पेसिफिक यही जीन है जो आपकी जो है ना ऑक्सीडेटिव स्ट्रेस को मेंटेन कर रहे हैं, या इसके अलावा बहुत सारे और भी जीन्स हैं?
इसके अलावा और भी बहुत सारे जीन्स हैं। The maintenance, as I said, it's a physiological process. Our body is basically armored with a range of defense systems. So it's basically just some of them. They are basically like multiple of them.
Thank you.
Okay, Dr. Tahsin, I have a question. What's good of finding out the oxidative stress level once the individual has been diagnosed with thalassemia? And the second part of the question is that, okay, the stress level is high, so what could be the antioxidative treatment for reducing the stress level?
Well, for the first part, the significance of identifying the oxidative stress level in the patient, that's what I'm understanding. So the significance of identifying is just like diagnosing or understanding a problem that is actually exists there. We have to first accept that the patients are experiencing high oxidative stress or high stress level, and only then we can think about curing them. The generation of ROS left untreated is one of the prime causes of the cancer generation in the body. It is one of the most primary leaders of multiple myelomas happening, right? So, and apart from that, it is responsible for malfunctional proteins and insufficient metabolic pathways, a range of them. Talking about how do we cure or how do we work around that, once we have identified, so we can not only check them for secondary complications, all of them associated with the increased oxidative stress, that is tissue fibrosis, as well as their chances of acquiring or basically the overexpression of proto-oncogenes, because I said it's going to be cancerous in a longer run. Regarding the treatment module of them, they need to be provided with, because they are provided with calcium supplements, vitamin D supplements and iron chelators. That is the most of them. They need to be provided with antioxidants as a part of their defense system. They have to be kept on a higher levels of vitamin C as a part of their non-enzymatic therapy mode. However, if suggested, they can be kept on a higher enzymatic mode of antioxidant therapy as well. There are plenty of drugs and treatments available for that.
And how does the polymorphism change the length of the protein? Because I guess it's only a replacement of a nucleotide in the
Yes, it is a replacement of the nucleotide because it replaces ACG with ATG, which is a start codon. So it starts the protein a little quicker, so it makes it a little longer. Yes. Was there someone else?
Yeah, thank you, Dr. Tahsin.
Thank you.
We have next presentation from Mr. Yasir Lakhani, Department of Biosciences, SZABIST.
ठीक है. Hello, can you guys hear me? Assalamu alaikum. My name is Yasir Hussain, and I'll be presenting my final project which I did for my bachelor's. This was with my group members Zahra and Hamza Javed, and our supervisor was and and my supervisor was Ma'am Rusa Ijaz. So the topic To introduce the topic, first I'll let you know that this is related to green technology. In green technology, we'll be specific about that we have created a process for dealing with hazardous substances in our waterways. We are a coastal city and we are very fortunate for that, but unfortunately due to rapid industrialization, we have been dumping a lot of toxic chemicals into waterways, which have caused a lot of issues with our food supply chain, in our environment, and in the overall public health scenario. So the presentation is entitled Immobilization of Fungi on Coconut Coir and its uses for bioremoval of malachite green. These are the contents of the presentation. We'll be going over the introduction. So malachite green is a green colored dye and it is used in many industries, specifically in the textile industry. It is toxic and carcinogenic in nature. The removal of it is therefore crucial. Since we have used fungi for this application, so the enzyme of interest we have is lacase, which is a fungal extracellular enzyme which can degrade the dye structure. This is the image. This is the image of the fungi of interest. Malachite can be degraded to leucomalachite green and conversion to N-dimethylated metabolites. The objective of the study was very simple. First, we get the matrix, we immobilize the fungi on the matrix. Immobilization is a process where we fix the structure of the fungi onto a matrix, onto a carrier, and then we perform the microremediation, and then we optimize the dye removal process. So the matrix we used for this application was coconut coir, and then we conducted the immobilization of fungi on coconut coir, which was a very simple process. Is that we incubated the coconut coir with the fungi of interest in broth culture, of course, and then we used the immobilized fungi for the bioremoval of malachite green. Then we performed the dye decolorization assay, and then statistical optimization, as that is one of the crucial important step. Whenever we create a process, we need something which is efficient so that we can replicate it. And then of course the immobilization process is an extrude added step, so we like to remove them, and we went ahead and performed the recyclability of the immobilized fungi for dye removal, and then the scanning electron microscopy. So the initial results and discussions were Traumatis pubensis, we did the enzymatic assays as well, produced 506.8 IU per liter, and this is the staining. This is the staining of the fungal culture with lactophenol blue, and the culture you can see here. So this is how the immobilization process can be visualized. Of course, this is just the initial photo. When we get deep into the scanning electron microscopy, the structures get more diverse and more dense with the matrix. Initially, to check if the decolorization was present, we did a decolorization assay, and these were the initial results from it. With the immobilized Traumatis pubensis MB89, which is the fungal of our interest, we were able to get 86.71% decolorization. We also used free cells of the fungi, which gave us 51.65% decolorization, and then we used the coconut coir, which gave us 64.66% decolorization. The initial data we generated from the decolorization assay was fed into Minitab software, on which we ran the central composite design, which gave us 16 initial experiments to run, and we worked on 3 factors: temperature, reaction time, and dye decolorization, and up to 5 different levels of the variations. This is the ANOVA of the central composite design. So the value of interest was less than 0.5, and those were only significant here, here, and here. And the Pareto chart is the graphical representation of this chart. This is the contour plot, and the contour plot signifies it was between reaction time and temperature, dye concentration and temperature, dye concentration and reaction time. On this side we have got the dye concentration, and on this side we have got the reaction time, which was in hours. And the darkest center is where we can see the greatest amount of decolorization in all of these plots. So this is the money shot photo. As you can see, this is how an optimized condition would look like. I'm not sure if you are able to see the dark green color right here, which you should, and this is how the process would actually degrade the malachite green dye into non-toxic compounds. And the optimized conditions were temperature was 30 degrees centigrade, reaction time was 59.27 hours, and dye concentration was 0.006%, and this resulted in 100% dye removal. We also check for the recyclability of immobilized fungi, because of course immobilization of the fungi is an added step. So what we did is we immobilized the fungi initially, and then we replicated the experiment of dye decolorization again and again with the optimized condition. So after running 12 cycles, we still had greater than 95% removal of malachite green from the solution. And these are the scanning electron microscopy images, which were quite interesting when we came to see them the first time. This is the one with the coconut coir. This is at 500 magnification. This is at 3500 magnification. This signifies that the coconut coir was used because you can see that the structure has many porous places, and this is the immobilized coir. You can see the hyphae structures interacting with the matrix, and this is at 500 magnification, and this is at 3500. This is a single hyphae strand magnified. So the conclusion is that bioremediation was an effective method to remove the toxic dye. We presented this in multiple international and national conferences. This was presented in the first biological research symposium organized by Aga Khan University. This was a poster presentation there. This was at the 13th International Biennial Conference organized by PSM. This was quite an experience to remember. This was also a poster presentation. We also presented this poster, this idea, rather the prototype at the DICE arranged by Dow University of Health Sciences. And then we presented it at Muhammad Ali Jinnah University as well as a poster presentation, and at the second International Conference on Biological Research and Applied Sciences. This was virtual. The abstract was also selected for the ASM microbe, which will be held in Washington DC 2022. So at the end, I would like to acknowledge SZABIST for providing me this platform to create such an innovative project, and I would also like to acknowledge the people who have worked with, my supervisor and my group members, and of course Dr. Mohammad Sohail, from whom we obtained the culture and who has been a constant support to us. He's from the Department of Microbiology, University of Karachi, and he has opened his lab doors to us like it's something we never expected to. So we are very grateful to him, and thank you so much.
Thank you, Yasir. It was a good presentation. So we have any questions from audience? Yes, please.
Hello, my name is Zurekha Faisal. I wanted to ask you a question. What properties do coconut coir have that enables it to immobilize fungi?
So whenever we look for a carrier, there are certain eligibility criteria we look for. Some of it is not actually based on the physical and chemical characteristics, but it is more relevant to the physical characteristics, but not so much to the chemical characteristics. So when we look for a carrier, we look for the how much can we get of it. Is it abundant? The second thing we look for, is it cheap? The third thing is we look for, since this is green technology, so we are looking for something which is degradable. There's one thing which you need to consider is the structure of the carrier, which is very important, such as if the carrier is porous, does the carrier has has an up and down surface area, that would be the appropriate word for. So these are some of the things which you need to consider while selecting a carrier. We use coconut coir because of the porosity of the substrate, and the second thing was that it was cheap, it was abundant, it was green.
Thank you.
Welcome.
Do we have more questions from audience? Okay, thank you very much, Yasir, for a wonderful presentation. Our next presenter is Mohammad Mubashir Khan from Dow College of Biotechnology, Dow Institute of Health Sciences. अच्छी स्ट्रेटजी होती है हाँ हम लोग धमाका करते हैं होने के लिए
Okay.
Assalamu alaikum and a very good afternoon all of you. My name is Mohammad Mubashir Khan. I am MPhil Biotechnology Scholar at Dow University of Health Sciences, and I am today presenting a part of my MPhil research which is entitled as Pathophysiology of Cardiorenal Syndrome in Beta Thalassemia Major Patients, a Molecular Insight. Moving on towards first we should know what cardiorenal syndrome is. It is basically a pathophysiological condition in which either of the organ, either of the heart or kidney dysfunction leads to the dysfunctioning or injury of the other organ. Cardiorenal syndrome develop with a number of clinical indications. One of them is fibrosis. Fibrosis is basically a condition of tissue rigidity which leads to cardiac or renal dysfunction, and this cardiac and renal dysfunction leads to the activation of various pathological systems like RAS signaling pathway, production of natriuretic peptides, production of reactive oxygen species and pro-inflammatory cytokines. Beta thalassemia patients are basically at higher risk of developing CRS because of their continuous blood transfusions, which leads to the iron overload in their body. Furthermore, oxidative stress also plays a very vital role in the production of, in the development of cardiorenal syndrome. These reactive oxygen species, as mentioned by Dr. Tehseen, leads to the activation of different pathological pathways like renin-angiotensin system signaling pathway.
This RAS signaling pathway leads to the production of an enzyme which is NADPH oxidase, and this enzyme overproduction leads to the overproduction of reactive oxygen species. These reactive oxygen species, when accumulate in our body, they causes DNA damage, they causes mitochondrial enzyme inactivation, they causes stand breaks and base hydroxylations, and all of these manifestations leads to cellular injury and apoptosis, ultimately leading to cardiac and renal aberrations. There are certain inflammatory markers that are that have been reported to be elevated in cardiorenal syndrome, which are NT-proBNP and creatinine, and they both are in NT-proBNP is basically the cellular health biomarker of heart, and creatinine for kidney. There are certain molecular markers, markers that regulate cellular health, which are AKT1, GRB2 and SIRT1. All of these biomarkers regulate cellular senescence, cellular health through AKT-mTOR signaling pathway. What beta thalassemia is? Beta thalassemia is basically a heritable disorder which is caused by a point mutation in beta globin gene. The global estimation, as you all have heard before, there are about 82% individuals who only survive by the age of 30. And if we talk about Pakistan, there are about 100,000 active cases of beta thalassemia in Pakistan. Whereas I have come across a paper which mentioned that in beta thalassemia patient, there is about the frequency of CRS is 27.8%. What happens in beta thalassemia patient? They basically are exposed to very high concentration of iron, and that high concentration of iron leads to myocardial fibrosis and other cardiac aberrations, as well as some renal aberrations as well. And both of these cardiac and renal aberrations leads to the coexistence of cardiac and renal injury, which is not primarily because of thalassemia, but it is secondary to thalassemia, and these injuries are classified as cardiorenal syndrome type 5. So beta thalassemia patients must need to be considered for secondary secondary pathologies, including cardiorenal syndrome, other than hemoglobin level associated conditions. Currently we have data available on CRS, but that data basically encompasses the diagnosis and therapeutic side. It's not about the modulation of cellular health and cytotoxicity. No study have been reported as yet which signifies inflammatory and oxidative stress associated with cellular senescence or cellular damage in cardiorenal syndrome. And this oxidative stress, inflammatory cytokines, and various other mechanisms, metabolic dysregulation leads to the comorbidities which are not primarily caused by beta thalassemia, but became the cause of death in those patients. So objectives of this study was to evaluate the risk of cardiorenal syndrome in beta thalassemia major patients through respective serum biomarkers, and also to analyze the expression of cellular metabolism markers associated with cardiorenal syndrome in beta thalassemia major patients. What we did, we collected blood samples from overall 200 subjects, 100 were healthy and patients each. The blood sample was used for RNA extraction and for serum separation. The RNA was extracted using triazole reagent method, and that RNA was quantified, treated with DNase enzyme, converted to cDNA, and then used for used for expression analysis by using qPCR. The serum separation was done by simple centrifugation, and that serum was used for NT-proBNP levels through ELISA and quantitative analysis of serum ferritin and creatinine as well. And all the data was subjected to statistical analysis using SPSS version 24. So these are the results. First of all, the biological parameters. Here is mentioned that beta thalassemia patients have a higher level of serum creatinine and serum NT-proBNP as compared to healthy individuals, which indicates they are much more prone to cardiac and renal injury as compared to healthy individuals. Also, there is a significant higher level of serum ferritin in beta thalassemia patients as compared to healthy individuals. which is basically mentioning the iron overload. This is the expression analysis data, and with the all the three biomarkers GRB2, AKT1 and SIRT1, they were analyzed for their expression in beta thalassemia patients and as well as in healthy individuals, and the expression was measured by using delta CT method, and all the expression were normalized using the housekeeping gene GAPDH. So GRB2 levels were significantly higher in beta thalassemia patients, and AKT1 and SIRT1 were lower in beta thalassemia patients. So what we conclude with this study, higher serum ferritin levels indicate iron overload in beta thalassemia major patients, which leads to tissue rigidity and organ failure. Higher serum anti-proBNP levels in beta thalassemia patient shows disrupted cardiac activity, and higher serum levels in beta thalassemia patients shows compromised renal health. Also, overexpressed GRB2 and underexpressed AKT1 and SIRT1 combinedly manifest some of the outcomes which are hypertrophy, fibrosis, mitochondrial dysfunction and inflammatory oxidative stress, and all of these conditions leads to cardiac and renal failure, and which basically manifests as cardiorenal syndrome. This study is still under continuation. Serum lipid profile is yet to be analyzed for the study subjects. Although the study helps us evaluate the risk factors in beta thalassemia patient, but we need to expand the sample size as well, and this preliminary data suggests that transfusion dependent beta thalassemia major patients, they need to be routinely analyzed for the cardiac and renal health biomarkers so that they can be considered as cardiorenal syndrome patients as well. Thank you.
Thank you, Mubashir, for such a nice presentation. So, if you guys having any question? No question? Okay, I have one question.
Yes, sir.
Was it possible to make a subcategory of thalassemic patients with CRS and without CRS, with established CRS?
Sir, we have not done it yet, but we have planned to do it. This study is still under continuation, and we are the data is still being subjected to statistical analysis. So we have planned to do that as well.
So I guess these serum markers and gene expressions level may serve as prognostic markers to guess whether the thalassemic patient is going to have CRS?
Yes.
Huh?
Just if the main question that gets into we cannot divide them. I think it should be fine. We cannot divide them before the analysis because they have not been diagnosed as CRS patients. It's like we cannot make groups now. We can only make the groups when we have completed the study and doing the final statistical evaluations. We cannot recruit the patients based on if they have CRS or not because they are not being diagnosed, at least in our system or in more than 90% of the beta thalassemia centers around the world. We know that they are at risk of CRS, but it's not a part of their routine evaluation, although plenty of them actually die of renal failure rather than based on that. So since they have not been diagnosed, we cannot make the groups now. So
I guess there must be some patients with thalassemia and with some renal.
Yes, of course, because not 100% of them did not have elevated proBNP levels. Neither 100% of them had creatinine ones. So, and of course, once we segregate of them, there would be some that would have higher proBNP and lower creatinine. So, a patient is only going to be classified as CRS if it has got an elevated level of both. So that would end up having 4 groups, like normalized for both, raised for either one, and then raised for both. And then we are going to divide the expression analysis in these 4 groups as well. But that can only be done once everything's all done.
So, Dr. do you think that you can go for the retrospective data, your patient who already have this, and you can go back and because you won't have.
We can, but the patients, their records do not have anti-proBNP levels. It's not the part of their routine tests. Some of them can have the creatinine levels, but it's not a routine thing, so it's going to be very few of them. That is one of the reasons when we contacted the thalassemia center, we checked it like which one of their routine tests that we can acquire and do not have to do it again, because that would be the repetition. So these are the ones that they just don't do.
this is what the hematologists from there actually convey to us, and as well they have identified, but still they do not do trop-I levels as well. They are subjective, they should be like trop-I levels should be considered for them as well if we are considering for cardiac hypertrophy, but it's not a part of their routine thing, so we cannot have the retrospective data.
Thank you very much.
So thank you, Mubashir, for a wonderful presentation. Our next presenter is Ms. Maria Azam from Department of Biochemistry, University of Karachi. इसके बाद last. एक नहीं आया, एक absent इसलिए हो रहा है, last one तो बहुत जल्द.
Loud. Assalamu alaikum. My name is Maria Azam and I'm from Biochemistry Department, University of Karachi. So here's basically my PhD work, mitigated effect of brown algae on CCL4 induced liver fibrosis. Okay, let's begin with the chronic liver diseases. Basically, chronic liver diseases, the global burden of chronic liver diseases increases day by day. It's maybe due to the consumption of drugs, maybe chemicals, and there's pathogens, several including the viruses and bacteria which are responsible to induce chronic liver diseases. The global burden, according to the CDC report, is around 2 million. I wrote over here as a 1.5, but it's approximately 2 million of the prevalence around the globe. Basically, what is liver fibrosis? Like my earlier one said, the fibrosis is a condition which induces the tissue hardening. So basically, what happened over here is liver fibrosis is a hardening of liver tissues due to the chemical or maybe any pathogens which is responsible to induce the liver fibrosis or the hardening of tissues. Initially, it was considered as irreversible, but nowadays it's considered as reversible because at initial level, liver fibrosis is caused. Sorry for this because it's my first time. Initially, what had happened that researcher was considered that only pro-inflammatory or anti-inflammatory cytokines are involved in the progression of the liver fibrosis, but here we are targeted the pro-inflammatory cytokines, which may, if we treat it on time, it may be reversed. So, okay, what happened? In my study, I used the tetrachloride, a chemical which is an environmental contaminant and used in many industries. So, what happened? When tetrachloride enters in our body, it activates the cytochrome P450 system, which ultimately provokes the activity of cytochrome 2E1, which leads toward the induction of ROS and ultimately induces the nuclear factor kappa pathways, which causes the activation of liver macrophages, basically M1 macrophages over here. And then M1 macrophages causes the activation of the pro-inflammatory cytokines which activate the hepatic stellate cells and convert into myofibroblast cells. And then it increases the deposition of collagen, collagen type 1 basically over here. I haven't mentioned it, but okay. And then what the theme was, basically we use marine macroalgae for the treatment of liver fibrosis. Why marine macroalgae? As our coastal areas is rich with the brown one. So we use the brown seaweed, the ethanolic extract of brown seaweed, this Phaeophyta, basically a brown seaweed. It contains the polysaccharides, phenolic compounds and fatty acids, which helps in attenuation of oxidative stress induced by the repeated administration of tetrachloride. And it also shows biological activity against hyperlipidemic, anti-cancer and antimicrobial and neuroprotective activity in previous reports. Okay, what the theme was, basically we targeted specifically the inhibition of inflammatory cytokines to attenuate or to reverse the liver fibrosis. Okay, we divided animal model into two groups. One was the fibrotic rat, and other one was the normal control rat. The fibrotic rat was treated with the 50% of tetrachloride dissolved in olive oil, and we administered it thrice a week for 30 days. And the similar goes with the other groups, like they are supplemented with the silymarin. Silymarin is over here used as a positive control. It's a herb and marketed as Silybum. And it's not basically it's not FDA approved, but it's used as a supplement. And here we use the Pediana pavonia and the Spatoglassum asperum, and we use the ethanolic extract of these seaweeds. And while the normal group was normally contained administered with the distilled water, and the group was only supplemented with the Pediana pavonia extracts and Spatoglassum asperum extract and the Silymarin extract. Okay, what happened, like here we prepared the ethanolic extract, and we also run the GCMS of our hexane fraction, and we induce the liver fibrosis by the administration of tetrachloride, like I told you before, and we administered the ethanolic extract. On day 31st, what we have done, we decapitated the rats and we obtained the blood, and we analyzed the serum markers, as well as we also analyzed their apoptotic protein concentration, and we administered their we also checked the interleukin level and histopathological evaluations, and we did the estimation of the inflammatory action and the tissue content of rose and reactive nitrogen species. And these are basically these are the primer we use, and the statistical analysis. So here my results are basically the supplementation of Pediana pavonia and asperum significantly reduced, as you can see over here. They significantly reduce their hepatic markers. They reduce the concentration of ALT, AST, LDH, and ALP as compared to our fibrotic group. Moving toward our total and direct blue ribbon level, they significantly reduce it, 74.57% and 56%. Okay, here is our interleukin 6 serum concentration and the caspase concentration. So here Pediana pavonia and asperum both reduce significantly. And these are the oxidative stress which is induced due to the CCl4. You don't have it. Okay, you can clearly see from here that asperum and P. pavonia significantly reduce the oxidative stress as compared to tetrachloride. And they also reduce the CCl4 reduce the GSH concentration, GSH protein concentration, while the asperum and P. pavonia markedly reduce the asperum tissue concentration of LPO and nitric oxide. I don't have the laser over here. Okay, these are the. Okay, here is the basically ethanolic extract effect on the GPX and the catalase activity. They significantly improve the CAT concentration and GPX concentration, the activity of antioxidant enzymes. And here is mRNA expression of the inflammatory mediator. You can clearly see from this. Okay, the tetrachloride increase the expression of the TGF beta 1 and also increase the expression of collagen 1, interleukin, INOS, and TNF alpha. These are the inflammatory markers we are targeted, and they also increase significantly increase, while the supplementation of both extract reduce markedly. And here's like here you can see the collagen expression increases in the tetrachloride, while the supplementation of Pediana pavonia and asperum extract reduce. So here you can see the Mason trichrome staining we have used. It increases the collagen deposition around the dice spaces. So this blue color, maybe you can see clearly, this blue color showed the excessive deposition of the collagen around the dice spaces, while the supplementation of asperum and P. pavonia reduces the deposition of the collagen around the dice spaces back to the normal, not exactly the normal, because there is a fibrous tissues you can clearly see, or maybe not collagen deposition decreases, but not that morphologically it's not that much significant, but it may be reverted if we go toward, or if we are planning to do more. So here's the Gcms analysis. Like I said, that I use the hexane fraction, what are the possible components which are responsible to reduce or attenuate the liver or revert the fibrosis. So here is a list, a very diverse list. I saw it's a decanoic acid, pentadecanoic, hexadecanoic acid. These are the fatty acids which are present in our seaweeds extract, or they may have role in attenuation of liver fibrosis. There are a couple of research papers reported that these are the components which may have antifibrotic activity. So here is my future perspective, that what I am going to do is the isolation through the HPLC, and we will check the protein expression and further pathological pathway which may involve in liver fibrosis, and our potential component maybe have role to attenuate it. Thank you so much.
Okay, very well presented presentation and in time. So if you have any questions from audience? We have no questions, okay. So as we are running out of time, so thank you so much for the wonderful presentation. Now we'll have our last presentation of this session, Ms. Iraj Javed from Department of Bioinformatics, Sir Syed University of Engineering and Technology. मुझसे बाद में मिलिएगा, कुछ क्वेश्चन है, नहीं कर सकता था इस वक्त, सेशन हो जाए ना, उससे कौन पूछेगा फिर? वहाँ पे नहीं पूछे ना, चाय भी पूछेंगे.
Right, और ये left. Hello, assalamu alaikum. Hello, assalamu alaikum. My name is Iraj Javed. I'm a final year student in Sir Syed University of Engineering and Technology at the Department of Bioinformatics. I did this project in my, Is it okay now? Can anyone hear me? Yes. I did this project in my genomic course, which is entitled as Antimicrobial Resistance of Naegleria Infections using Whole Genome Sequencing and Molecular Timing of Protozoan Naegleria. Let's start off with the introductory part. Now this is the outline of my presentation. We're going to discuss the introduction, the methodology section, the results and discussion, the conclusion, and the future perspective. So let's start off with the introductory part. As you have seen that Naegleria fowleri has been a widespread in all over Pakistan since the past few seasons. So it's basically a species of amoeba or a brain-eating amoeba that lives in warm fresh water like hot lakes, rivers, springs, muddy soils. And you know, it is said to be a brain-eating amoeba because it mainly affects the human nervous system. You know, it was first discovered in Australia in 1965, but was later to be evolved in the US also and other states of the countries that are facing severe hot climatic conditions. And due to these hot climatic conditions, they were said to be thermophilic in nature because they can survive or withstand temperatures of about 114 degrees Fahrenheit, which is according to Celsius is 46 degrees or 45 degrees. So it mainly infects the human nervous system. And how does it do that? You know, the active swimmers are a big target of this infection. When they go for swimming and when the water reaches their nose, as you can see from the diagram, that when the water reaches their nose, the olfactory nerve releases some chemicals. They get attracted to those chemicals and they transport them to the frontal lobe of the brain, where they destroy those meningeal membrane cells, or causing them inflammation and leading to meningoencephalitis. Now, Naegleria was widespread in the past few seasons. We know that, but it was due to the water contamination being widespread in Pakistan. And you see the pools, the water, the lakes that were not properly chlorinated. So mainly the rural areas were affected with this because, you know, they were facing severe poverty conditions, and due to these poverty conditions, they only received the groundwater, and the groundwater consists of all the microbes and the infections. And so there are several antibiotics and antifungals being given to kill those Naegleria or fowlery infections, but somehow for the human case it's quite difficult. So what we set in our research objective was to conduct a comparative genomic analysis of this Naegleria fowlery to compare it with the other organisms, and to see whether they are highly similar to the other organisms, and evaluate to see if these strains have developed some resistance genes or not. So let's move on to the methodology section. This is a research flow chart of our methodology. The first part, what we did is identified the research problem. Then we did a little background research about the infection. What were the causes? What were the consequences of this infection? Then we used our genomic data through different bioinformatic tools to analyze their similarities and evaluate the prediction on our results. So let's start off with the data collection part. Now, you know, in order to work on any genomic data, we have to retrieve it from somewhere. So we use NCBI, which is the National Center for Biological Information. It's basically a curated database that consists of data from all different data resources or biological resources. So what we did was we placed in organism's name or ID, and we to which we get a lot of sequences, and we selected the Naegleria fowlery genomic sequence. Now what we did was we input the accession number. You can either put the FASTA file, it doesn't matter, and then we run the BLAST analysis from which we get this results as I will show you the table here. So you see each of the percentage identity are about 80 to 90%, which means these strains that we have obtained from our results are about highly or mostly closely related to the query sequence or the genomic sequence that we have entered. So so you know we need these things in order to build a phylogenetic tree. So after what we did was we used the Mega software to create a phylogenetic tree, but before that what we did was we aligned it by cluster W. Now this is the Mega software. We have opened the align FASTA file from which we have downloaded from the BLAST. We have downloaded this, we have selected specific strains, and we have downloaded from the BLAST, and we opened this file in the Mega software. This is the alignment file that is being shown. So now what we did was we select this alignment option and aligned it by cluster W. Now the reason for using cluster W was cluster W is basically a machine learning algorithm. It's an algorithm that is designed on the back end of the software to create multiple sequence alignment and generate a phylogenetic tree. So what we see in our results and discussion session is that each of these strains of Naegleria, according to the phylogenetic comparison, are mostly closely related and are highly similar to one another, to each of the other pairs also, and with itself with the pairs also. So after what we did was that we used PubMLST to compare this aligned FASTA file with the other organism, because we wanted to do the comparison with the other organism. So what we did was we input the sequence or the FASTA file here, and we get this result. We can see mostly gaps in the alignment, and mostly aligned, mostly are the gaps. So the gaps reveal that they are not much closely related to the organisms sequences present in this database. Because you know PubMLST is a database for consisting all the organisms and other species ID, like different types of bacteria, fungi. You can get any organism sequences here. So we compared it and we get this kind of results to show that they reveal mostly gaps that which reveal that our results that Naegleria strains are not much closely related to other organisms. So after what we did was we used ResFinder tool to find the antimicrobial resistance among this Naegleria strain. So rest finder tool works just like a BLAST tool. Unlike BLAST, uses to find higher similarity among the NCBI database, which consists of all the sequences of different organisms. But what rest finder tool does is it finds the highest similarity among the resistance genes present in this database. So what we did when we import our FASTA file, we get this kind of results which show that this is the antimicrobial agents, this is their domain, and they show no resistance. This no resistance means that the resistance genes that were getting resistant to these kind of drugs were not present in the Naegleria strains. So that's why so we can conclude here that we did a little comparison of the recent genomic findings, and antimicrobial drugs are extremely important in killing pathogens and destroying those cells, and they are all used in all modern biotechnology, but somehow they develop resistance, and due to this resistance are either developing mutations in the future. We can predict that if Naegleria does develop this mutation in the future due to some resistance gene, due to these resistance genes being present, then we must devise some preventions and approaches against it. Like what we can do is we can control the global warming areas that have been altering the distribution of Naegleria. We can prescribe different drugs to see whether resistance genes will be present in the future, and still it remains to continue to be a big threat, and a global collaborative effort is needed for all types of technology to work on this. So these are the references. I would like to express my gratitude to Sir Syed University of Engineering and Technology and my teachers and supervisors, Ms. Rabia Fazan, and the Department of Bioinformatics for giving me this opportunity to work on this great initiative, and which has evolved me to do my extensive, an extensive research and build my new knowledge and skills. Thank you.
Thank you, Raj. It was a very good presentation. So we are again open for the questions. Do you have questions? Iraj, would you mind giving answers to questions?
Sorry. Any questions?
Okay, that was really interesting. My question to you is how does global warming play any role in this?
As I've told you in the introductory part, that due to high temperatures, global warming is occurring due to, and it's causing high temperatures. So if there are high temperatures, then this will give rise to the Naegleria infection, because they can survive temperature of almost 46 and higher than that. That's why global warming is altering the distribution of Naegleria infection, because through this the temperature gets rise and they will eventually evolve.
Thank you so much.
Welcome.
Okay. In your study, you compare Naegleria with some other microorganisms. So what is the purpose? Like, we do know that Naegleria is a protozoa, and if we compare with some other organism, so there are some differences. So what is the purpose?
You see, if I'm designing a drug for one bacteria organism that has a genome that is most closely related or it's destroying, that drug can be used to destroy this genome. So if that genome has a similar to Naegleria strain, so that drug can also be used for Naegleria. So that's why we did the comparison. Yes, and the comparison was being done by the phylogenetic.
And can you explain more about what is rest finder?
What is?
Rest finder.
Rest finder is basically a tool. It's a BLAST tool. It works like a BLAST tool. You see, in order to find the highest similarity, it has some resistance. Like every database has some genes in it, all types of genes. This is a special tool. It also consists of many sequences, but also resistance genes present in them. So it compared those resistance genes with this. So that's why it's a useful tool if you come to use of it.
Basically precise selection के लिए उन्होंने
Yes.
Okay.
For finding high similarity among sequences.
Okay, Rich, I have one question.
Yes.
So, as you mentioned that there was no resistance with some of the antibiotics, those were vancomycin, etc. And it means that this is very obvious and the clinicians are neglecting it, number one. Either this is the case, or this is not always the thing that if you see by a bioinformatic tool that something has resistance or not, you actually need to perform experiments.
Yes.
So there's not always the genetics, but some epigenetics and other factors are also involved. Your comments? I mean, you show that vancomycin resistance is not there, and it
Yes,
means that
but,
Naegleria can be killed by vancomycin, right?
Yes.
You are saying this.
No, these bioinformatic tool does produce results, but we cannot be accurate about it. We have to go for further research, right? This was just generation. Like it's something that is being said, and it's like we have seen, but somehow we have to do further research. What we can do is right now, after this, we can do molecular docking and using bioinformatics, virtual skin and molecular docking to see whether the drugs have the resistance genes can be used, or the drugs can be applied to those Naegleria strain to defeat those Naegleria.
Again, molecular docking is again the computerized,
yes,
right?
It's a computer.
So you have to go to the bench side also and see whether
Yes.
these things are working or not. Sorry?
Yes.
Yeah, this is prediction, but this is going to give some result when you combine this thing with the lab data, right? Inshallah. Fine. Thank you so much. It was again a very good presentation. So with this, we come to the end of this, what session it was, the research session, scientific session, and again back to the host. Naam nahi pata, I'm sorry.
Thank you, researchers. Thank you, researchers, for such informative presentations. We will now commence the distribution of shields, for which I request Dr. Kashif Ali to make their way towards the stage. And present the shields to our respected chair, co-chair, and presenters. So please come to stage as you hear your name. Dr. Sayyid Zulfikar Ali Naqvi. Dr. Yasir Raza. Now our presenters, Dr. Tehseen Quds. Dr. Mumtaz Hussain. Fabiha Naseem. Dr. Afan Shoaib. Muhammad Wajiuddin. Sana Minai. Dr. Tehseen Fatma. Yasir Lakhani. Mohammad Mubashir Khan. Maria Azam. Irad Javed.
Okay, now I would like Dr. Kashif to present the shields to our MOC for today's ceremony. Farba Abbas. And Shamir Ahmed.
Lastly, I am very honored and delighted to deliver the concluding remarks for the first international conference of life sciences, LIFESCICON, held at SZABIST Karachi. Thank you for your presence, speakers, guests, faculty, and students. I hope that you have enjoyed and learned the insights and opportunities throughout the conference and will take advantage of it. Therefore, let me wish us all a lot of energy, enthusiasm, and commitment towards a future for all. Thank you so much. Allah Hafiz. मेरी पिक्चर नहीं आई 2 मिनट दो खुल गई अच्छी आई है लेना लेना Please.